from yesterday, and now we'll start. Mine is going to be very low tech, just to get some concepts down and maybe as a segue to the other speakers. But I'll start by saying that HTE, heterogeneity, is never zero. And so I think testing is irrelevant, not only here, but I must admit almost every place. Estimation is the key, but the point estimate really doesn't tell the whole story. One needs to have some idea of uncertainty and more generally of the range of the underlying parameters. And being at least labeled a Bayesian, I think the ideal approach is to get the full posterior distribution of the between subgroup variance component and of individual effects, and use that full distribution to make policy or clinical or whatever the decisions are. It's going to generally require knowing the uncertainties in a broad range. If you're a frequentist, do confidence intervals, but maybe I'll propose more than just the 95 and so on. Give at least a depiction of the full histogram, if you'd like. And then in the same spirit, make decisions based on what you found in the data relative to things like quantitative or qualitative HTE, therapeutic windows where you're trading off side effects and other kinds of negatives. There may be different criteria for the regulatory context, the clinical context, and indeed we have snapshots for patient communication. Each of these might require a somewhat different analysis and a somewhat different decision-making based on the analysis. The main point is, and it's certainly a theme from yesterday, and I'm sure for tomorrow, and indeed for the last 40 or 50 years, is you need sufficient stability to make any progress. And usually the direct estimates are too unstable. Trials are not really designed for stability of direct estimates, at least at a very refined subgroup level. So here's my little statement about stability without having it move around in real time, but at least there it is. And so you need some form of stabilization. And pretty much any approach you take is going to trade off subgroup specific focus for estimation stability, trading off bias and variance. And there are a variety of ways of doing that. And I'm not by any means listing all of them, but aggregation, you can just pool. All the way, if you'd like, to the overall effect. Pooling will stabilize, but it loses focus on the building blocks. Regression in machine learning stabilizes, but generally gives you a model that pays, other than having built itself, no attention to the specific direct estimate. And then Bayesian hierarchical modeling, or hierarchical modeling in general, which tries to borrow information within and amongst data sources, not just necessarily the data in the current study, to maintain some focus and at the same time stabilize. So here's my kind of cartoon version of that without getting very formulaic. It's really the variance bias trade-off that's the theme of a lot of what we do in statistics, whether it be Bayes or otherwise. You want to estimate an attribute, in this case a subgroup effect or a set of them. And you have some direct estimate, maybe it's unbiased, maybe not, but at least it's delivered directly by the data context that you're focusing on, want to focus on. But you also have a model that may be a regression model, it may be machine learning, it may be actually just data from some other source, possibly more stable than the direct estimate. And you want to combine those two. And at least one criterion for doing well, or for measuring how well you did, is the expected squared deviation between the true value and what you predict. You could replace the 2 by some other letter if you'd like, but this one is the one that produces the nice mantra of variance plus squared bias. And with that in mind, you can compute a weighted average of the direct estimate and the model, whatever you're thinking of as the generic form of the model, with a weighted 1 minus D and D. If you're a Bayesian, we usually do D and B and 1 minus B, but I thought I'd make it data here. You can regroup that to be model plus some weight times the residual. And so the idea is to use the model and then given a certain amount of weight to the residual of the data from the model, where the amount of weight you're going to do is computed by the relative mean squared error of the model divided by the sum of that and the direct estimate. And you've got to be able to estimate these. The variances are usually pretty easy to estimate, the biases not so. But the main idea is that if the mean squared error for the data increases, and usually it's just the variance of the data, it's usually unbiased, then D decreases and you rely on the model more than the direct estimate. But as the precision of the direct estimate increases, then D increases and you give more weight on the data, reducing the reliance on the model. And a Bayesian hierarchical model is very effective, not the only way, but a very effective way of implementing this, especially to come up with some way of getting the MSE for the model. So here's an example, not from the clinical thing, because it's a pretty picture, if nothing else. These is malaria prevalence in small areas in Zambia. And these are the direct estimates, and there's a little toggle I could do to spread these out, but this is the direct estimate. These whiskers are proportional to the width of the Bayesian credible interval, and longer whiskers meaning more uncertainty. This shrinkage is to a pretty rich regression model with things like proximity to rivers, so on and so forth in it. But the bottom picture is kind of the typical Bayes picture. These are now the residuals from the regression model, raw and now shrunken. And you can see, at least in this case, the direct estimates are relatively unstable. There's a lot of shrinkage towards the common value. This would be like shrinkage towards the overall effect in a clinical trial. And in this case, not much reliance on the direct estimates, mostly on the regression. But if we were to increase the sample size of each of these small area estimates, you would find that there'd be less shrinkage going down here. It would happen automatically, basically. So a few clinical examples. This is a golden oldie. I use old examples partially because I am, but also to show that nothing here is radically new. Some technology is, but the idea certainly isn't. This is hypertension detection and follow-up program. Randomized to referred care, which you go to your clinician, you've got high blood pressure, versus stepped care, which is a sequence of curation, if you'd like, interventions. Take the 5-year death rate and looking at 12 strata or 12 subgroups. The direct estimate is 1,000 times the 1,000 there to keep away from decimal points, the odds ratio. I, in the chat yesterday, said I'd rather use clinically relevant risk differences, but I'm going to stick with this for now. And here's the direct estimate of it, not necessarily perfectly unbiased, but at least direct. And here are the strata of blood pressure, cross-tabulated with race and gender. And I won't give you the numbers, I'll just give you a plot. As with the malaria case, here are the direct estimates. I've left the whiskers off just to make it a little cleaner to look at. High blood pressure, white male, medium white female, high white female. These were also the smaller N groups. And here are the Bayesian posterior means, or if you'd like the weighted average, like the one I had on the previous slide or two, showing that at least if you were to believe the Bayes posteriors, all of the point estimates are showing benefit. I've put down here something partially to stress the flexibility of the Bayesian approach. You could have a goal different from optimally estimating each subgroup. You could have the goal that I call ensembles, which is that you'd like the collection of estimates to be a good picture for the collection of true underlying values, called the ensemble, or a good distribution estimate. They turn out to be less condensed than the posterior means, less spread than the middle ones. They're kind of a Goldilocks solution. And if you believe these numbers, I happen to, it looks like as a point estimate, there are 2 groups that are truly showing lack of benefit, 2 out of the 12 in the distributional sense. Now it turns out that, at least back then, and I think it's probably still true, it wasn't so much that step care was bad, it was that referred care was pretty good for these people. Not only they had good clinical context, but they behaved well, if you'd like. And so which of these is the truth? We don't know exactly, but this has some pretty good statistical properties and is worth considering compared to this one. Another one that probably Steve should be the one actually talking on this, and that he was the statistician on it, involved in it directly. But it was to look at treatment for congestive heart failure and low ejection fraction. And there were lots of people randomized, pretty amazingly good, with only an N of 1 difference. And you had 452 deaths in the treatment group and 510 in the placebo group. Here are the probabilities of death, the hazard ratio, and the log hazard ratio, showing an overall benefit, obviously. So let's look at this one in terms of, well, it just happens to be 12 subgroups again. Gender by age, by baseline ejection fraction, some of them looking, I would say, depressingly low. Well, all of them depressingly low. And here's a picture from, by the way, you can look at these references to see the kinds of details that are going on in this. Here's a typical forest plot, solid line, the overall effect, subgroup by subgroup examples. And at least here, it's not just binary cuts. It's at least a relatively fine-grained division and partition that may reduce a little bit the threat of confounding. And you can see the direct estimates and their confidence interval, which are in black. The ones that are really broad are the ones obviously with a smaller sample size. They get shrunken much more radically than the ones that are quite precise. Shrunken both in the sense of the width of the confidence interval and the red dot at the center. They obviously go towards the overall effect. There's not a strong signal for any kind of subgroup effects here that are at least worth having some dramatically different clinical opinion about. A side comment here is that one thing you might want to consider is forcing the red dots, when you take their weighted average relative to these sample sizes, to actually line up exactly with the solid black line. In the survey world, that's called benchmarking. It's the idea of making sure to induce credibility, if you'd like, that when you're all said and done, instead of just optimizing each subgroup, do an end game that makes sure that the overall result, weighted result, if you'd like, is compatible with the overall trial result. If your model is quite good to begin with, it will be a very minor adjustment. If your model ain't so good, then not only will it be a major adjustment, but it will be a strong signal that something's wrong and that you probably need to do better. Here's another advertisement for Bayesian flexibility. I've drawn here a picture of what would be the true underlying treatment effect. And one of the things I like to push, whether it be for frequentist or Bayes, is let's have discussions about what we would do in the context of true underlying effects. Let the data be a window on it, but yes, I want to see your data, but I'd actually really want to see your inferences back to the nether world where in fact you never really get to see it perfectly. So here's a picture for just 2 subgroups. You could make it for 10 subgroups and somebody better than I could draw it or use a graphics program to do it. The next session may be able to do it. So I've done it as though greater than 0 is a win and less than 0 is not. So here's a region in terms of the parameters of clinical effect for the 2 different subgroups where it's a win-win, both of them win. Here's lose-lose. Here's a case where there's a qualitative interaction where there's a win for subgroup 2, but a loss for subgroup 1. And yet in this region, the overall effect will still be a win. In other words, the weighted average here will be enough to bring things over to the positive side. Here, however, not true. And ditto for down here for the other thing. You could decorate this if you'd like with side effects and have if you'd like you better have a really good treatment effect to compensate for the negative effects, just like Stuart Pocock did yesterday, and you could build that into these regions. My main point is that these are hardly rectangular. It's very challenging to think about how you would even compute something in a frequentist mode, subject to having a prior distribution, and it's in the supplementary slides, but it's not necessarily completely trivial, but it's doable. You can compute posterior probability of any of these regions or any other regions you'd like, and then make decisions on the basis of those probabilities, or more generally on where the high density stuff is in these regions. So for me, another promotion of Bayes, not mindless promotion, but at least a promotion, is that you get to do things that I think are either impossible or at least very kludgy by any other method. So shrinkage, and maybe Bayes more generally, can be controversial. It is controversial. Highly uncertain estimates are substantially shrunken towards the model and potentially hiding the truth. I've done several years ago a lot of work on hospital profiling, and one of the big complaints there is, God, these lousy hospitals that are small sample size get shrunken all the way back towards the former mean and hides their malfeasance, if you'd like. On the other hand, the ones that are looking good hide their benefits. It's a common criticism, and it's not one without merit. However, we commonly rely on regression estimates. We mindlessly do, I shouldn't say mindlessly, we happily do regression. We also commonly rely on direct estimates. So why should sort of the middle ground be so controversial? And I think there are a lot of reasons, not all of them listed here. And one is lack of familiarity with and trust of the approach. Two is a lot of times it's only exposure to the basic formulation where the shrinkage is to a constant, to an overall number rather than to a well-constructed model. Now in the clinical trials context, it might be very logical only to shrink towards the overall treatment effect. But if you have a very few well-considered covariates, whether they be gender or otherwise, it's worth considering putting those into the target. So I think a lot of the criticism, and a lot of it that's up here, is that the shrinkage is towards a common value as opposed to towards a reasonably well-constructed regression model. Concerns about Bayesian modeling, and you can go back even further, but the 1986 paper by Efron with discussion to me is still an important read for almost everyone. It's not technical, but it's got Brad's comments, Dennis Lindley, many other people to just frame the issues, I think, today just as well. There are a lot of books on Bayes. I've reframed listing ours. It's now ancient history almost, although I think everything still in it is true. But Gary Rosner and colleagues have a book that I'm not sure is actually out yet, but will be out pretty soon, and has a very serious attention not only to Bayesian technology, but to advice on using Bayes. So I think there's a lot of reason to carefully consider Bayesian methods, but not mindlessly. Going back to this for a minute, this is kind of the traditional partitioning into these well-defined subgroups. The limiting case of this is to partition into every patient, to have a risk score for them, to have a profoundly active Bayesian approach that will allow stabilization at the patient level. That's a work in progress, but I think is sort of the way to go eventually. So one more slide and I'll get out of your way. Well, two. One says thank you. I'll let you know before I get to it. I'd like to propose that we all anticipate the need for stabilization. Collect attributes, covariates that inform the shrinkage target, the model. Maybe traditional ones, but also maybe some more laboratory values or whatever might be relevant to the context. Harmonize with past and prospectively harmonize with future studies. Collect attributes that may help take this study's findings and dock them to another finding by weighted averaging and so on and so forth. Make sure data definitions get aligned, including the covariate definitions. Document things such as censoring policy. It's one of those that is usually, I shouldn't say usually, but frequently left out. Is no news or is no news good news? That kind of thing can really determine the follow-up time for a person. Serve the commons by anticipating future uses. And in the regulatory context, obviously there are others much better skilled to do this than I, but I've been hanging around the FDA for a few years anyway. Look at the impact story that was mentioned yesterday. You still have to have a trusted and transparent protocol and process for developing prior distributions, developing the model. Hopefully we do that for whatever approach you're using, making decisions and so on. The particulars are going to be different from the frequentist criteria that are currently in place, but the goals are the same. And so I think the idea of not getting away from transparency and not getting away from a priori specification, but moving to a different mode of thought is itself a work in progress, but one that has been done quite well in the device center, but needs to be, I think, infiltrating a bit more into the other centers. The Bayesian approach is very effective. I am not a mindless proponent of it. It's not a panacea. It requires a lot of care. Herman Rubin was once quoted as saying, a good Bayesian beats a frequentist every time, but a bad Bayesian gets clobbered. You've got to be careful. And also traditional values apply. Space age techniques will not rescue Stone Age data. There's nothing that's going to rescue a poor study. So that's it. I'll let others take over.
Thank you, Tom. That was great. Let's move on to the next speaker. So,
I'll stop share and then somebody else.
Great. Don Berry is founder of Berry Consultants, LLC, and professor in the Department of Biostatistics, the University of Texas MD Anderson Cancer Center. Dr. Berry is one of the most prominent statisticians in the field. He's probably best known for his work on Bayesian adaptive trials in cancer and other diseases, and in particular for his design of the landmark I-SPY 2 trial in high-risk early breast cancer. Oh, Dr. Berry.
Thanks, Steve. I'm not sharing. I'm just going to say next. Is that right? Or should I share?
No, that's right. That's
okay. Right. So next, please. My disclosure, co-owner of Berry Consultants, and I'll be talking about two examples that Berry Consultants advise the clients. Next. Subgroup analysis is an epidemic in drug and device development. It's dangerous. There are landmines everywhere, but they're essential. It's precision medicine. The story of drug development is and will continue to be looking at who benefits from the therapies. And so homing in on subgroups is really essential. But retrospective subgroups are almost always wrong. And there are horrible examples of people following, chasing wild geese. I hope that these two are not part of those. The two presentations are actual presentations at FDA panels on a tale of two catheters. One is a hot catheter and the other is a cold catheter in two different diseases. The presentations that I'll give is almost exclusively the slides that were presented to the panels, and I haven't adulterated them, but I'm not going to talk about all aspects of them. So next. The first is the ELAIR system. Next. It's a trial prospectively designed 2-to-1 looking at patients who had severe asthma. And this device goes into the lungs and heats up the various pathways and reduces the muscle so that the patient can breathe better. It was wholly Bayesian designed. The sample size was finally set at 300 with 2 interim analyses. The objective was to show that the device was superior, and I'll tell you the endpoint in a minute, with 95% probability of superiority, and then to continue to the follow-up for 12 months for each patient. Next. The endpoint was AQ-LQ, the asthma quality of life questionnaire, and the comparison was to sham. Next. These are the data, the results of the trial, showing the change from baseline at 3 months up to 12 months. And you see that the ELAIR across the approximately 300 patients showed a benefit of 2:1 in the change in baseline in AQ-LQ. Next. The FDA had questions about the analysis, and in particular, the regional issue, the geographic issue. And they were concerned about the population of the United States, not surprisingly, and what the data in other countries had to do with the United States and what to believe. They wanted to use the data in its totality, but focus on the United States population. Next. So this is the bar chart showing the United States, Brazil on the right, and other US, other OUS countries. And you'll notice that the left two bars show a benefit for a layer. The Brazil bars, however, show the opposite. Next. Of course, one wants to look at the differences between Brazil and the United States, and it turned out that Brazil had a rather different population. It was basically less severe asthma. That's what the first bullets are showing. And they had different levels of treatment of concomitant care. Next. What I showed, this of course was a big deal at the panel meeting, and the focus was on Brazil and how it related to, and did it relate to, and should you use Brazil? I mean, that was the primary goal. The primary endpoint was to be analyzed across all of the studies. So I showed this geographic representation using a Bayesian hierarchical model, looking at the various regions and saying, how do you combine these? Next. So this is a picture to represent differently what Tom was discussing. This is a population of regions, and these 5 regions that are named were sampled. There are some that weren't named. We didn't look at China, for example. So a critical parameter in the Bayesian hierarchical analysis approach is the heterogeneity, the sort of variance in this population. What's the hierarchy? The hierarchy is the patients within the regions and the regions themselves. The experimental units are either one of those, and indeed, there can be several levels of a hierarchy. You can look at a continent, for example, and regard the countries within the continent as being still another level of the hierarchy. Next. There's a hyperparameter, tau, and one usually in a Bayesian approach has a distribution on that, but I want to show you the effect depending on tau. Next. So what is tau? It's large if the various countries, regions have great differences in terms of the treatment effect. It's small, next, please, meaning that there's a little heterogeneity. So in the small case, you want to borrow a lot. In the large case, you don't want to borrow much because they're sort of standing alone in the extreme. Next. So this is a picture. The endpoint is on the vertical axis. The countries and regions are on the horizontal axis. The United States had most of the patients, followed closely by Brazil, which had 29%, to Australia that had 5%. If tau is 0, if there's no variability at all, then you do the pooling. This is the primary analysis of the primary endpoint. And the probability that Alera was better in this poolability case is 96%. Next. If you go to next, if you go to the other extreme, where there's great heterogeneity, and assume that the individual regions are independent trials, completely, no borrowing at all, no shrinkage at all, which of course is wrong. Tom said it's wrong, I agree. Both of these are wrong. Maybe something in between is right. And that's what I did. I showed this following movie. Next. So this is a modest value of 0.1. This is showing picture with a different set of pictures, what Tom was showing. If tau is 0.1, then there's some borrowing. Next. And just go through the whole thing till you come to tau equals infinity. Now, pay attention to that leftmost graph, that leftmost box. That's showing the mean in the US and showing the variability in the US. And now, and there are two things happening. One is that as you're borrowing less, the mean gets bigger and converges to what the actual US mean was. But the box gets wider. So those two things are characteristic of the hierarchical approach. And go back and do the movie again very quickly. Just go back to the earlier thing and then forward again. Just go through quickly. And the circle at the bottom, the oval at the bottom, shows that throughout the entire range of tau, no matter how much borrowing there is, the US population estimate is beneficial. And next, that was enough for the panel and for the FDA. Next, that they approved asthmatics, and asthmatics was sold to Boston Scientific. Next. The other one is the Thermocool, which is for paroxysmal atrial fibrillation. Next. Is a randomized trial, again, 2-to-1. Next. The primary goals were to address the effectiveness of this Thermocool catheter ablation in preventing the recurrence of atrial fibrillation. The comparison was the catheter to antiarrhythmic drug therapy. The effectiveness window was 9 months. Next. This is the timeline, and just go through the timeline quickly. The sponsor had difficulty accruing patients, and eventually came to us and said maybe we could convert to Bayesian. Patients like Bayesian. No, I'm kidding. And maybe we'll accrue better. And indeed, that happened for a number of reasons. And we worked with the FDA to get interim analyses in and doing a Bayesian analysis. Next. 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 Oops, too far. The leftmost of these are the sites. The leftmost is OUS1, which is a site in Italy. And it was the biggest site. It had 30% of the patients. Next. The company argued that the results should be pooled. Next. The statistical analyses, of course, prospectively defined and the type 1 error assessed using simulations, etc. Next. There were 160 subjects that were enrolled out of a total possible of 230. The protocols did prediction of individual patients depending on what their early results were. The result of the first interim analysis, the predictive probability of success overall was 0.999, and you'll see why that is. And early success was declared. Next. This was the Kaplan-Meier plot. The primary endpoint was at 9 months. You see it's 64% for Thermocool, 16% for antiarrhythmic drugs. Next. And these are the densities for that those two values, published in JAMA eventually. Next. The success rates and the failure rates of the two therapies, showing that there are some that are still, that were censored in that Kaplan-Meier plot. Next. The FDA and the panel meeting was all about regional variation. Not all about, at least half was this one site in Italy, the OUS1. Next. It's gone. What happened? Can you hear me?
Yes, we can hear you.
Oh, okay. So these are the non-US sites. There are 95. Next. These are the US sites, less dramatic, 44% at 9 months versus 18% at 9 months. Next. This is the OUS1 site. There were 31 patients assigned to Thermocool. All of them were successes. So none of them had recurrent AF. The AAD arm had about half that, had actually 18 patients, and 11% were successes. When I saw this, I said, this is an Italian site. I said to the company, is this in the Vatican? I mean, how can this be? So the company, the FDA, was worried about this. You know, what is going on here? And so this meeting next, these are the remaining sites next. This meeting was about understanding how the OUS1 site could be so different and so dramatically favorable. And there were differences. It turns out this investigator was very experienced with this device and was, you know, had a great record. Anyway, I think the bottom line here was that since the US sites showed a benefit, a significant benefit, they would approve. Next. Next. So they granted market approval and it's still marketed. It's, the device has changed over time. They've improved it over time. So two stories next, two tales of two catheters, both successful, both addressing this regional differences using Bayesian hierarchical models, and were at least in one sense successful. So thank you, that's from me.
Thanks very much, Don. We'll move on to the next speaker, Gene Pinello is a mathematical statistician and researcher at the FDA CDRH. Gene's a fellow of American Statistical Association, past chair of ASA section on medical devices and diagnostics, current president of the FDA Statistical Association. I think Dr. Pinello will be referring to his book chapter on Bayesian subgroup analysis with hierarchical models that he co-authored with Mark Rothman. And I'll just add that I found that chapter to be really helpful as part of my education on the topic. So, Gene.
Thank you, Steve. And I guess you'll advance the slides, right?
Yes.
Everybody can hear me? Okay.
Yep, can hear you.
Yeah, great. So I want to thank the organizers and Steve Snappin for this session, but especially Hayat Ahmed and Barbara Beer, because this program is terrific, and the wonderful lineup of speakers. I mean, that's no small feat. So I really appreciate all the hard efforts that they made to develop this program and this conference. So what I want to discuss is, well, my aim today, and we've already heard some wonderful presentations on Bayesian hierarchical models, which can be a mystery to many folks. And my aim today is to unravel the mystery a little bit more. And there'll probably be some redundancy to the previous presentations, but redundancy is not such a bad thing. But so what I'm going to do is I'm going to compare the Bayesian hierarchical models for one-way and multi-way subgroup analysis with the separate frequentist types of analyses of subgroups. And I'll compare them on point estimates, interval estimates, and decision-making, hypothesis testing, in an attempt to make the contrast clearer. And to simplify the comparisons, I'll assume some of the parameters of the models are known. So in this slide, this is a figure from Gelman et al. At the bottom there, but compares treatment effects across 8 sites, and I'm not going to get into the details of the example, but the left panel are the treatment effects and confidence intervals within the 8 sites unadjusted for multiplicity. And the middle panel shows separate analyses of the sites with a Bonferroni adjustment, which is the typical frequentist analysis, or at least one procedure you could use. And the right panel is the Bayesian hierarchical model in the paper. Could you, next slide, please? And you can see that comparing the left and middle panels, that what's happening here is the Bonferroni correction. What's not happening is the treatment effect estimates are exactly the same. The frequentist approach with Bonferroni correction does not adjust the treatment effect estimates at all. They remain the same, and that's what I'm trying to depict here with the horizontal line. But we know, and from the previous presentations, we know that the sample estimates of the treatment effects within subgroups tend to vary too much because they include the true treatment effect variation plus random variation due to finite sampling variability. So we know that the estimates probably have too much variation relative to the true treatment effects. Thus, that makes the separate analyses of the treatment effects within subgroups difficult to interpret statistically and clinically. The right panel is the Bayesian hierarchical model employed in the paper, and you can see that the sample estimates are shrunk toward the overall estimate, which is the red dashed line. And it creates an ensemble of shrinkage estimates that more closely resemble the variation of the true treatment effects, or actually a little bit more shrunk relative to the true treatment effects, as Tom Lewis described. Next slide, please. And with the red dashed line here, I'm comparing the lower limits of the confidence intervals and for the third site, which the treatment effect seems to be particularly low. But to compare the approaches, the middle panel shows the the Bonferroni correction. And what's happening here is you just expand the confidence intervals. And why do you do that? Is because you want to control the overall probability of a falsely significant result. But in stark contrast, the Bayesian hierarchical model actually narrows the confidence intervals relative to the left panel, which is the unadjusted analysis. And that why? Because the shrinkage estimates for the subgroups borrow information from all the subgroups. And as has been described, it depends, the amount of borrowing that takes place depends on the variation within the subgroup compared to the variation between subgroups. Next slide. And I want to give an example. Actually, Steve Snappin yesterday talked about this. The study of losartan versus atenolol on MACE events in patients with hypertension. And 80% of the trial was consisted of non-Black patients, and 20% were Black patients. Next slide. And this is a list of the sample size and person years and events in the subgroups of non-Blacks and Blacks, and overall for each of the treatments. And you can see the event percents and the event rates, and for the point estimates of the event rates within subgroups show benefit of losartan in the non-Blacks, but potential harm in Blacks, the opposite direction, as you can see in the rightmost column there. Next slide, please. And this is the hazard ratio analysis. Overall, there is a benefit of losartan, and it's statistically significant according to a frequentist analysis. The confidence interval on the hazard ratio is completely below 1. Two-sided p-value is below 0.025. Next slide. So in non-Blacks, you get about the same values for the hazard ratio and confidence interval because most of the data are in non-Blacks. So F is is the frequentist analysis. FB is a fully Bayes hierarchical model analysis, and you can see there's not a whole lot of difference in the point estimate for the hazard ratio in the middle column and the 95% interval estimate. And so there is not much borrowing going on here because the variation within non-Black subgroup is quite small compared to the variation between the subgroups. Next slide. But among Blacks, because the sample size is much smaller and the person years are much smaller, you do get a shrinkage from 1.67, which is, in the opposite direction for the frequentist analysis or the sample or maximum likelihood estimate. But to the fully Bayes shrinkage estimate of 1.52, and the interval estimate covers 1. So it in the fully Bayes analysis from 0.93 to 1.47. So there is some uncertainty here of whether losartan may be harmful in Blacks or not. According to this analysis, if you agree with, or if you think the hierarchical model is a reasonable model to fit to this data. Next slide. So what I want to do is go through some formulas, so forget the notation here, but I think it's pretty simple to understand. And I want to go through a one-way hierarchical model and a two-way hierarchical model for subgroup analysis. Next slide. Okay, so in a one-way subgroup analysis, we treat all the subgroups as exchangeable. What I'm going to do here, this is called a normal model. I'm going to start with the sample estimates, which are these vector of Y's of J subgroups that have, these are the sample estimates of the treatment effects, which are the mu's, and they have variances given, these are the sigmas. And the model is that the the sample treatment effects are normally distributed about the true treatment effects with the variances sigma j squared. And with the Bayesian hierarchical model, you assume that the true treatment effects are exchangeable, that they're or any ordering of them is equally plausible. And one way to implement that is to assume that the treatment effects are independent and have a common distribution, given in 10.2 here, with a mean mu naught and a variance sigma mu squared. And sigma mu squared represents the variation between the subgroups and the true treatment effects. Next slide. And so with Bayesian analysis, you want to look at the posterior distribution of the true treatment effects given the data. And the mean here is, which is in yellow in 10.3, is a weighted average of the mu naught, which is the population mean of the true treatment effects, and the sample estimate yj. And the weight has to do with, the weights have to do with this factor Sj. It's a shrinkage factor, which is 1 minus 1 over phi j, and phi j is this, measures this variation between the subgroups versus variation within. It can be considered the true F ratio in an analysis of variance. And you can also see that in blue, the posterior variance is the sample variance, or not the sample variance, but the variance on the sample estimate sigma j squared times the shrinkage factor. So this is why the confidence interval, or the interval estimates in the Bayesian analysis get narrowed compared to the unadjusted separate analyses. Next slide. And so these are the Bayesian credible intervals compared to the frequentist confidence intervals we did separate analyses. And I'm trying to make, so in blue are the differences. So you see that there's a weighted average plus or minus standard normal deviate, that's z 1 minus alpha over 2 times the posterior standard deviation, which is that square root of Sj times sigma j. And so what's in blue is the difference compared to the frequentist confidence interval. So I think that shows the difference. Next slide. And, I think when we see forest plots and where we look at the traditional standard analyses of forest plots of treatment effects within subgroups, but I think we ought to be asking as well, are there actual differences between the subgroups in the treatment effects? And so this is an analysis of the differences between subgroups 1 and 2, according to this model. And so 10.6 gives the posterior mean and posterior variance for the difference, which I'm calling delta 1, 2, between the two treatment effects. Now, if it happens that the variances are exactly the same, the sigma j squared equal a common value sigma y, then this simplifies to the the posterior mean being shrinkage factor S times the sample difference D1, 2, and the posterior variation being S times the variance on the sample difference sigma d squared. So it's a very simple formula. Next slide. And so that leads to the interval estimate being on the difference being this formula here, which is the shrinkage estimate SD plus or minus the standard normal deviate times the square root of S sigma d. And so the two values in blue show the difference between this and the normal frequentist confidence interval. Next, please. And so you can also do decision analysis. So in Bayesian methods, you would declare the difference to be greater than 0 if the posterior probability is greater than some value, and I'm calling it 1 minus alpha here. And what that leads to is that a critical region for declaring the difference being greater than 0 being this Z12, which is a standardized difference, standardizing the sample difference divided by its, by dividing by its standard deviation. And that has to be greater than that standard normal deviate, the Z1 minus alpha over 2 times the square root of the shrinkage factor. And that could be, so you can see the comparison here of that critical region with the standard frequentist critical region, which is just the standardized difference being greater than the standard normal deviate. So you can see that depending on the shrinkage factor, you can get much more conservative inference or not much more conservative inference. And it all depends on that variation between subgroups to the variation within. Next slide. If you wanted to be what's so-called empirical Bayes, you could estimate what that shrinkage factor ought to be and just plug it in. And I was telling you before that it really depends on this F ratio. So the true F ratio, well, you can just substitute the observed F ratio and then analysis of variance. And this is what the critical region would look like. This only makes sense if the F ratio is greater than 1, which it doesn't have to be, but sometimes these empirical Bayes regions are very good approximations to the fully Bayes empirical regions. And the next slide shows that if you only had 2 subgroups, the F ratio turns out to be the square of Z12, so you could plug that in assuming it's greater than 1. Next slide. And you know, I've been assuming previously that the variances are known within the subgroups, but you can do, instead of standardizing the difference, you can studentize it by an estimate of the variance, and you get these critical T values instead of the Z values, and you can see that they depend smoothly on the F ratio. And if the F ratio is small, you can get you have much larger critical T values to compare against the T statistic. Next slide. And for 3 subgroups, the F ratio is can be written as the sum of the square of 2 contrasts between the subgroups. I just wanted to show you could actually represent the critical regions for all pairwise differences of the 3 subgroups in 2-dimensional space according to those contrasts. And the dashed line show the standard frequentist critical regions, and the solid lines show the Bayesian hierarchical model critical regions. And in the middle there, the underscore notation means that you can't declare any pairwise differences to be any different, any of the subgroups to be different from each other, because all 3 subgroups are underlined, and you kind of get but that's exactly where the F ratio is small, and so you get this kind of distended hexagon region where you can't declare any differences. Expanded hexagon, because of the F ratio is small, the inference becomes more conservative. I'm not sure how much time I have left, Steve, but
I think actually we're running a bit over, so I would say not much time.
okay. So should I just go to the conclusions, or?
Yeah, I'd say, yeah, please do.
Okay, maybe, so I'll just skip all the 2-way hierarchical model stuff, but one remark here is that, and I think you can see from what I presented that the Bayesian analysis with the hierarchical model is kind of like a smooth version of a one-way analysis of variance, where in a one-way ANOVA, if the F ratio is insignificant, then you declare all the subgroup means or treatment effects are declared equal. It's kind of like the Fisher's LSD rules. And what I didn't present here is that that for a 2-way, you can build more structure into the subgroups where you could have factors like age, race, sex, or other factors, and build a model, as Tom Lewis was saying, where you don't just shrink to a constant, but you shrink in much more elaborate ways that make intuitive sense. And so the next slide is just saying that in the multi-way ANOVA, the multi-way analysis of subgroups is kind of like a smooth version of multi-way analysis of variance. And but the key that on the next slide is, you have to assume that exchangeability at some level. And in the example that I gave, the big question is in the life study, are blacks considered exchangeable with non-blacks a priori, or if not, are there some covariates available that you can adjust for so that the residuals can be considered exchangeable? I'll stop. Thank you for allowing me to present, and I hope that was helpful. And I want to thank all those folks who helped me prepare the presentation.
Oh, so I apologize. I was muted, and so I'll introduce Frank again. Dr. Bretz is a distinguished quantitative research scientist at Novartis. He's supported the methodological development in various areas of pharmaceutical statistics, including dose finding, estimands, multiple comparisons, and adaptive designs. Dr. Bretz's best known work, at least to me, is on a very clever graphical approach for multiple comparisons procedures. Frank.
Okay, thanks. Thanks, Steve, and many thanks to the organizers for inviting me and having me on this session in particular. So my talk is largely based on a joint work with Mohamed Olosh and Mohak, both from FDA, and Ralph Agostino, a work that was published in form of a tutorial paper a couple of years ago. Next slide, please. I believe that we are running a little bit behind time schedule, so I will try to be brief. So let me just briefly then indicate that for the purpose of that paper, we were distinguishing between three categories of subgroup analysis: exploratory, supportive, and inferential analysis. And having clarity on the type of subgroup analysis certainly helps to understand the different requirements, and in particular the different statistical methodologies that are needed. So again, I will be a bit brief. In the paper, we discussed supportive analysis, which look at investigating the consistency of treatment effects across subgroups once the efficacy has been established in its intended overall population. So such consistency of treatment effects would indicate robustness of the overall trial findings with respect to then discussing the applicability of the treatment to the overall population. But we also discussed and connected supportive analysis with inferential analysis that aim at establishing treatment efficacy in a predefined targeted subgroup and or in the overall population. So in this presentation, I will focus mostly on the inferential analysis, and if you just please go one slide and just another one, and talk about a few points to consider when at the design stage, when considering such a situation or such a setting. And to set the scene, consider a hypothetical asthma trial with the objective of establishing efficacy in the overall population and a pre-specified targeted subgroup. We assume that the targeted subgroup is defined through patients with a biomarker positive status, and the prevalent rate is about 20%. So as I said, in the following slides, I will just briefly discuss a few points that need to be considered at the design stage, but also making the connection to the type of supportive analysis that I had briefly mentioned on the previous slide. So next slide, please. So one important aspect to consider, at least in inferential trials and for inferential subgroup analysis, is to consider multiplicity. So multiplicity can take different forms, and whether you are Bayesian or frequentist, I guess it just shows up in one way or another. And certainly, if you look into treatment effect estimates, you probably want to adjust for the treatment effect estimates in a smart way. And we just heard a couple of ways using Bayesian shrinkage estimates, I think, which is a great way to do it. Now, if you want to do a significance test for the treatment effect in an overall study population and a pre-specified targeted subgroup, then at least in confirmatory trials, you probably want to adjust in a suitable way for the multiplicity, and that would be needed. And one way of doing this is the graphical approaches that Steve had kindly mentioned. And these kind of graphical approaches allow you to visualize your testing strategies. And one such visualization you see on the right-hand side at the bottom. So you see different nodes. And if you just focus on the top two nodes, so these are the formal hypothesis. On the left-hand side would be HO, so this would be the node for indicating that you would not have any treatment effect in the overall population. And on the right-hand side, HT would then be that you would have no effect in the targeted subgroup, so T for targeted subgroup. And you also see that these nodes as associated with certain weights, which would kind of which would kind of help you to identify how you weigh these different objectives for your clinical trial. In particular, they may help you finding an importance relationship between the hypotheses. So essentially, you're splitting the significance levels between the nodes, between the hypotheses, and the edges between the nodes help you in recycling significance levels once you have been able to reject a null hypothesis, so that you don't lose that significance level. So that leads to stepwise test procedures, that can be visualized by these graphical approaches, and these visualizations do cover standard cases such as the ordinary sequential testing, hierarchical testing, and Bonferroni procedures, but also more advanced gatekeeping procedures, including the fallback procedure. Now, what you also see is the node at the lower left. So this is not a hypothesis, but this is an additional consideration that needs to be taken care of at the design stage, that when you have to think about decisions on the population for treatment use. So this leads to further considerations about the complementary subgroup, not in terms of a formal test, but still you need to think about suitable thresholds to establish a consistency across your subgroups. So this is summarized by the informal node on the lower left, and I will talk a bit about this in a few minutes. But before I move on to the next slide, maybe just to say that this methodology that you're seeing here, with the help of visualizing these testing strategies, they can be applied to more complex situations like group sequential designs, adaptive designs, and you can also easily take correlations into account between the test statistics, because the correlation would typically be of a structural nature and you can calculate it analytically. So improvements are possible beyond the basic graph that you're seeing here. Now, please move on to the next slide. So this is just to indicate here, coming back to the different test strategies that can be visualized, and as driven by the choice of the weights in the graph that you have seen. So different choices lead to different power and sample size requirements, as you can see in the top right in the table. So what you see here in the 5 different rows are different test strategies and the impact on the sample size and power for the overall population and the predefined subgroup. I will not go through the individual numbers and the individual results just because of the, because of the shortage of time. But let me just quickly mention, the various methods so that you get a basic idea of the flexibility of the testing strategies, but also the importance of having appropriate discussions within the teams at the design stage about the relative importance of your clinical trial objectives and the weighting schemes that you want to apply, because you can end up with very different test strategies based on, team discussions. So the first two rows, row 1 and 2, are different hierarchical test procedures or sequential testing schemes, where you would test the overall population first, and if significant, then continue testing the subgroup. And the two rows essentially differ with respect to the desired power that you're trying to achieve. So whether you want to focus and study and ensure that your study power is targeted by or is driven by the overall population or the subgroup. Row 3 is the simple Bonferroni approach, which typically I would not suggest in this way, how it shows here, as it can easily be improved. But then rows 4 and 5 are about these fallback procedures that I mentioned before, which allow this recycling of the significance level. And the difference between rows 4 and 5 is that in the latter one, and so in row number 5, it also uses an enriched population. So if, as a sponsor, you are interested in an enrichment strategy to recruit more patients from the targeted subpopulation, then then the natural prevalence would allow, then you can do that in order to increase the study power. So that is just another design idea that you can play with at when designing the trial. So understanding such operating characteristics at the design stage is certainly important, and they form the basis to discuss different test strategies. And these 5 test strategies are really just 5 examples. Others could be used, and it is critical to select the order of the hypothesis test and for the overall population and the subgroup, as well as the relative weights for establishing the efficacy in the treatment in each of these 2 populations, which will in turn impact the allocation of the overall significance level. Now, at this design stage, there will certainly be many other factors that needs to be considered, and which I'm kind of ignoring here. But the one aspect that I would like to emphasize is, I think it's almost my last slide. If you go to the next slide, please, is the important aspects around determining the population for treatment use. So this should also be done at the design stage, where you think ahead how to set consistency thresholds to facilitate the determination of the population for treatment use at the analysis stage. That is, consideration should be given on setting such thresholds on treatment effects for the subgroups to be included in the population for treatment use. And this is where I will have to refer to the backup slides for more details. So I wasn't sure whether I would have enough time, so I left it for the backup slides. But as part of the supportive analysis that I briefly introduced at the beginning of my presentation, we reviewed and investigated different consistency thresholds that have been published in the literature, like the harmony criteria from Gary Cook, and another one, a consistency threshold for Mohamed Halosh himself, that all come with statistical and additional clinical consideration how to set up such thresholds, but not in a formal hypothesis testing framework, but to understand the relative effect of the complementary group with vis-a-vis the effect in the overall population and the targeted subpopulation. So, and these considerations at the design stage should be such that you're investigating outcome scenarios at the planning stage to guide the choice of such thresholds. And this would give you possibilities to come up with alternative ways to investigate the population for treatment use, depending on the potential outcomes, possible outcomes that you can expect at the trial end. So you see a little flow chart here on algorithm. It's just one way of doing this at the bottom of the slide. So you would, for example, at the trial end, consider the overall population only if the null hypothesis for the overall population is rejected and the complement subgroup does meet its threshold, but the one for the target subgroup does not. And you continue with the other similar considerations using this kind of flow diagram. And it's important to understand the operating characteristics as you go through this flow diagram when you discuss the suitable choices for your consistency thresholds, which in this flow diagram is denoted by this alpha sub C star. But again, it's not related to hypothesis testing. It's really taking clinical and other statistical considerations into account. But I guess I will have to defer to the paper for further details. So I guess on the next slide, please, I already at the conclusions, and maybe for the sake of time, let me just drop the conclusions here, because I think most of that I have said already. So back to you, Steve.
Great, thank you very much, Frank. So as I said before, we have time for some discussion, question and answer, and we welcome questions from the audience during this period for the speakers in this morning's session, or from speakers from yesterday's sessions as well. So before I do that, I just thought maybe I'd give the first opportunity to this morning's panelists if they had anything to add or to comment on the other presentations from this morning. Anybody on the panel want to jump in? If not, we did get one question or maybe a couple of questions from the in the chat, and maybe I'll read one of them to you. I would appreciate if the panelists could comment on the choice of prior for the variance component, i.e. variance between subgroup effects. This is perhaps the most critical parameter in Bayesian subgroup models. It seems to me that this would be more important in fully exchangeable models compared to regression-based shrinkage models.
This is Tom. I'll make a brief comment and then let others. It's a good question. Ravi, you always ask good questions, and frequently you even have good answers, but at least I'll try to do something. It's a problem, especially with a small number of subgroups. If you have a large number of them and at least reasonable information on each, you can be empirical Bayes or almost. But in general, for the number of subgroups we're talking about, or even a large number when there's very little data for each, you need some form of informative prior. And ideally, but also frequently, I think you can import some information from other studies. Not so much on the endpoint necessarily, but on some aspect of a between unit, in this case, subgroup variance component. But short of that, I think, at least not necessarily it's the end game, but Don's approach of just doing a slider and watching when it really matters can be extremely informative. I'll close by simply saying it's a good question, but you don't even get to ask that question unless you have formulated a problem where that issue comes up, and it's important that it comes up.
This is Don, and as I indicated in my note, yeah, I agree with what Tom says and what Rob says. This is a very important issue. I mean, time doesn't do justice to letting us flesh it all out. There are implications on things, and I mentioned type 1 error and power. There are implications on those quantities, and the regulators sometimes worry about this. I mean, always worry about it. The issue of being prospective. So if you're going to be prospective in the design of whatever you're doing, as Jean indicated, looking at, and as Frank indicated, looking at various endpoints and analyses, and adjusting for multiple comparisons, whatever, you may not be able to take the Bayesian approach because the impact on type 1 error can be very substantial. And so this is, and what does perspective mean? Does it mean, you know, as Tom has said on other occasions, Are you going to adjust type 1 error for everything you do in your life? So it's, I'll end with what Tom said. This is a very important question. It is not insurmountable, however. I mean, I'm optimistic about the use of hierarchical models in not only this area, but many others.
Thanks.
Yeah, I think I'd like to comment. I agree with what's been said, and as Tom said, you're probably better off using a Bayesian hierarchical model and having to deal with that question than not use it at all. It also depends a little bit on perspective what your aim is, and you could do various different priors or different fixed values. One of the aims I've been involved a bit in is improving subgroup estimated treatment effects. And when I think about the prior for the variance, as I think about the raw estimates being sort of the starting point. So I'm sort of okay being a little conservative on the distribution for tau squared, understanding that it might be under borrowing what might be optimal, but I'll err on the side of under borrowing as opposed to err on the side of over borrowing.
This is Gene.
Oh, go ahead. Gene?
Well, I thought I'd briefly mention that there's a Jeffreys prior, as has been discussed in the classic book by Box and Chow, and it's not what you might think it is. I mean, if you write down exactly what Jeffreys prior is, it's kind of a combination of the within variances and the between variance, and it tends to work. It can work fairly well. I don't think people use it very much, and also, and I might get criticized for this, but a fiducial approach can actually work well. And I know fiducial in many circles is dead, but Xiaoli Meng, I think, and maybe others have started this regular meeting, the Bayes Fiducial Frequentist Best Friends Forever conference that I know they've had several times, or they've actually started to investigate and reinvigorate the fiducial approach, because it does have some theoretically good properties. I know there's a non-uniqueness part of it, but that's a avant-garde kind of view.
Ravi, were you wanting to say something?
Oh, no, I just wanted to thank, I appreciate the response. And yeah, we have considered half normal and Jeffreys priors and done what Don was basically suggesting, try different values for the hyperparameter and just present the forest plots with all that. I think Tom may have showed one such plot as well in his presentation, but I just wanted to kind of ask this question because it always comes up no matter what we do, people are not happy with the choice that you make.
So Ravi, you can make their choice. I mean, you can consider, everybody's prior and have a get together and see if there is any kind of community prior. Or if not, the Bayesian approach is inherently subjective and different people will draw different conclusions because they have different prior distributions. So consider everybody's prior. When you publish, consider lots of priors to accommodate whatever the audience has interest in.
So Steve?
Yes.
If there's a little bit of time, the question just came in a few minutes ago from the Q&A about the exchangeability issue.
So I think that'd be
for a little discussion on that or not?
I do. I'm just going to yield to Barbara to tell us when to stop, but I think it'd be great to spend.
Why don't we continue with that one thought and then we'll take a break.
Okay, great.
I will, if okay, I'll make a brief comment.
You can take what time you need to explain it, since all of us would love that.
there's this Sturm and Drang and complete concern about the exchangeability assumption in Bayesian analysis, not totally misplaced, but you should have equal energy for the exchangeability issue in regular old-fashioned regression. I mean, the implicit assumption there is the same. So we need to pay attention to it. It's not exchangeability of the model necessarily that shrink towards a model that has only an intercept. The whole idea is to build a model in the prior so that the residuals are at least approximately exchangeable. You'll never succeed perfectly. All models are wrong, but some are blah, blah, blah. So I'm not trying to say we don't need to be concerned about it, but it is just not any more important there than it is in traditional modeling, and that means it's pretty important. We never get it right. Stein's rule and other things show that if you get it approximately right, you can do a very good job on estimation. And so I want to close by saying, I'm not saying forget about it, but I'm saying don't obsess about it any more than you would for other models, and start thinking with porting that same concern to your regression models. That's all.
We appreciate the comment. I don't know if anybody else wanted to comment on exactly that point before we go to break.
So this is Don. There's a lot of confusion about exchangeability. When somebody says exchangeable in this area, one is not saying that the patients from one of the areas, say we're talking about regional, from one of the areas is exchangeable with the patients in another area. So it's not at the patient level that we're saying exchangeable, but it's at the level, usually we should be talking about the exchangeability of the studies, at least partial exchangeability. And anyway, I've found that when one says exchangeable, one sometimes hears, oh, but they're not exchangeable, so we can't do any of this.
Okay. I would just add a question for you. If there were, are there methods that provide similar methods that provide shrinkage estimates that don't make an exchangeability assumption, or could it be possible to develop such a method? Would that be an improvement?
Well, Steve, yes and no. I think implicit in all of them is that there's at least enough parental control. What I mean is that things are coming from a higher level and are related, are siblings, but it is in residual space that that thinking needs to be done. If it's a linear model, it's an additive space, but in other models, it's a dimension. I don't think there's any approach that doesn't implicitly depend on it being reasonable to bring things together at some focus, but I just will repeat myself only because I think it needs to be repeated. It's in the residual domain that this needs to be discussed, not before you build a model.
Great. So, okay, thanks very much. Barbara, I think we're ready for break.
Yes, we are. And thank you all for a very informative discussion, and really one that extended our thinking. I look forward to resuming at 10:15. You can all stay on, and we'll just go off, mute your video and mute and stop your video, and we'll resume at 10:15 with the visualization panel. Thank you. I'm wondering if we should begin again. I am nothing if but a timekeeper.
Barbara, can you hear me? This is
I
Frank.
can. I
Great.
can.
Do you want to go ahead and let me share?
Certainly. Hayat, will you give Frank? And let me introduce Marcia Levenstein, who will introduce the panel while Frank is getting set to share his screen.
Good.
Marcia?
And Marcia, I'm assuming you don't have a slide you want to show.
I think that was it. But Marcia, do you have a slide other than that?
No, I don't have a slide.
Good.
You can hear me, right?
Yes,
You
we
know,
can. We can.
I don't have a slide.
Good.
The
Go
opening
ahead.
section slide. Okay, so we've had great discussion this morning and yesterday of the statistical and analytical approaches to investigating heterogeneity of treatment effects, primarily through the use of subgroups. We've heard about frequentist and Bayesian methods, but people often view visualization as distinct from analysis. They really are all part of an overarching approach to understanding and investigating heterogeneity of treatment effects. In this session, we're going to hear about data visualization and the role of graphical representation within this overarching approach. We have three wonderful speakers. The first speaker is Frank Harrell. He's a fellow of the American Statistical Association. He received his PhD from the University of North Carolina and then went on to teach at Duke for many years. Subsequently, he founded the Department of Biostatistics and Epidemiology at the University of Virginia School of Medicine and went on to be a founding chair and professor of biostatistics at the Vanderbilt University Medical College. Currently, he is a associate editor of Statistics in Medicine. He's a scientific advisor for the Board of Science Translational Medicine. He's the editor and board on the American Heart Journal and has been a longtime consultant for the FDA. While at the FDA, he's been a member of the several advisory committees, both cardiorenal and the rheumatology advisory committee. He also was lead of the Support Scientific Center and Analytical Center. And currently he is a consultant in many, many areas for the FDA and with other important companies. So I'd like to introduce Frank Harrell.
Marcia, thank you very much. And I assume everyone has this slide up on their screen. This is the links to two blog articles that I'm drawing from for the material that I'll be showing you. So you may want to write down these URLs. They both start with fharrell.com/post. I'm going to be presenting some graphical methods that you may not have seen, but I'm going to also sneak in a lot of statistical ideas in this session. I hope that's okay, Marcia. And I did want to say I had high expectations about this meeting, and my expectations have been exceeded. I've really been enjoying this tremendously, and I'm in amazing company. So I'm really thankful to be here. So this first article is really, it has some graphics in it and a lot of ideas about looking at heterogeneity of treatment effect. And I want to take a little friendly issue with something a couple of the speakers have said, is that heterogeneity is always there. I don't believe that's the case. And I'll show you an example where we have strong evidence against heterogeneity. And there's some meta-analyses in the literature showing anti-heterogeneity of treatment effect. In other words, less heterogeneity of treatment effect than you would expect by chance for certain domains. But I'm going to look at this in a couple of different ways, and I'm going to frame this in terms of a kind of a fully worked out example from a very large clinical trial, which is the GUSTO-1 study. So this is a study of acute myocardial infarction that Rob Califf led, and it's a very important study. This main comparison is with TPA versus streptokinase for patients with acute MI, and the outcome is 30-day mortality. So it's a very rich data set because we don't usually have 40,000 patient clinical trials, but when you have something that big, you can really look at differential treatment effect, and you can look at risk-benefit trade-offs and a whole lot of other things. And so the data set has some really high quality data, and including a beautiful age range, 19 years to 108 years, actually 110-year-old was in the study. So there was no age restriction like a lot of clinical trials have. And I think that's a real beautiful thing. And so we have some great covariate distributions. So the first thing, as Eval Steyerberg and others have done in the literature, is to develop a risk model where we factor in an additive treatment effect on the log odds scale. And then we're going to get into differential treatment effect shortly. But we developed this risk model, which has regression coefficients for what you're used to seeing. We have 3 treatment groups in this analysis. So there's 2 variables for treatment, streptokinase and TPA. We have age, kilo class, systolic blood pressure, and a variety of other variables. But then we're going to get into a semi-saturated model. So what a semi-saturated model is one that allows the odds ratio to not be constant with respect to any of the covariates in the model. So you see it symbolically, and I might just enlarge that a little. Day 30 mortality is our outcome variable, and we have treatment asterisk. That means treatment is interacting with all of these things. So that is a semi-saturated model that allows for everything to interact with treatment. So we have tons of opportunities for differential treatment effect, which I think is a better name than heterogeneity of treatment effect. That's another story. And then when we look at that model and we do a likelihood ratio test comparing the base risk model that doesn't have any interactions with the interaction semi-saturated model, you see that the likelihood ratio chi-square is 16.5 on 20 degrees of freedom, and the p-value is 0.68. So you would say that we really don't have much evidence at all against the supposition that the odds ratio is constant. In other words, there's no heterogeneity of treatment effect with regard to these covariates. And then if you were to look at the AIC, which is a measure of which model is better likely to cross-validate and have improved predictions, you see that the AIC is smaller for the simple model as compared to the fuller model. So that is telling you that allowing for differential treatment effect is worse than not allowing it for it, because it creates overfitting that is not offset by signal. And so we're finding more noise than signal in looking at the interactions. And then we can also look at this a different way, which is what is a measure of relative explained variation. And if you took the variance of the predicted values with the full model that allowed for interaction compared to the variance of the simplified model for the predictions, it's a nice simple measure of explained variation. You can see that the model that had no interactions in it is 0.996 as good as the model that allowed for interaction. So the interactions adds 0.004 of predicted information. But we're going to be graphing that to look at that in more detail. But the next thing we do is really to try to bridge the previous discussion. Well, several speakers have addressed Bayesian shrinkage. And in the frequentist world, our sort of poor man's approximation to Bayesian shrinkage is penalized maximum likelihood estimation. So this is really very, very similar to empirical Bayes. And it is computationally really easy to do, so that's why we do it. But a full Bayesian analysis would be much better. So what happens when you do penalized maximum likelihood estimation? I'm going to say, let's don't penalize the main effects in the model. And so those are going to have a penalty of 0. And then the interaction effects are going to have an increasing amount of penalty, which is going to be up to a penalty of 1,000 times 1,000 or 1,000,000. So what that means is like having a single variance for random effects, and we're not customizing the borrowing of information according to the covariates, but we're assuming a common borrowing for all the covariates. Whereas if you were doing a full Bayesian analysis, this would be a non-hierarchical Bayesian analysis. borrow different amounts of information according to your prior knowledge about the importance of the covariates for giving you a different effect of, say, the TPA treatment. And so we have only one penalty term. And if you look at the penalty terms for the interaction and the effective degrees of freedom that you get from that model, you can see if you go to a penalty of 1 million, you've penalized the model down to 12.03 degrees of freedom, whereas the main effects model with no interaction at all has 12.0 degrees of freedom. So you're really ignoring the interaction. Now, AIC is phrased here so that bigger is better. That's the reverse of the way we usually do it. And you can see that if you just follow this out, the optimum penalty is going to be infinity. So consistent with what we saw with AIC, it's best to completely ignore the interactions. But if you say, well, we're going to give the interactions one more benefit of the doubt and give it a penalty of 30,000, that means the interactions are allowed to have 1 degree of freedom total. So we go from 12 to 13 degrees of freedom with this model that allows for differential treatment effect. And let's look at the consequences of that. So we see that our coefficients for main effects, like what's the main effect of age, overall treatment groups, the coefficients of the main effects are almost unchanged in this penalized model, but the coefficients of the interaction effects are changed a lot. So this is the unpenalized range of the coefficient. You see they're penalized very, very close to 0, and that is giving them, instead of 20 degrees of freedom, that's giving them 1 degree of freedom. And then we could look at distributions of various effects from that formulation, and this is where we started to get into graphics. So the first thing you notice is when you have the probability of death under the control treatment, streptokinase, and then you have the probability of death estimated from this logistic model under TPA, that's really one of the best ways to look at it, because if you say, what's our risk of dying if you're control versus new treatment, we're not really making any statement to the patient should they care about odds ratio, or risk difference. It's all here. And you can see that that's not too far off the line of identity. So treatment is not your biggest risk factor. But we can also look at difference, absolute difference. And when you show the distribution of the absolute risk reduction due to TPA, you see this histogram that is incredibly spread and very skewed. And so you have a lot of patients that have an estimated benefit of virtually 0. These are patients that have an estimated baseline risk of virtually 0. But you have people getting a benefit all the way out to, say, about 0.08, but a very few of them. And so the absolute risk reduction of 1% in the Gusto study, that 1% is driven by these people way out here. So they're really outliers. And the 1% risk reduction, which is here, is not really a good measure of central tendency for your risk reduction. That's why I don't like using risk difference as a summary for a study. The median risk difference is 0.007, which is much more representative of what patients are likely to experience. But if you look at the distribution of the relative risk, you see it's fairly spread out here. And then if you look at and of course, this was the distribution of absolute risk reduction, which is very spread out. And then if you look at the distribution of odds ratios, you can see that it is a very narrow distribution around the overall point estimate you would get if you did not have any interactions in the model. So in interpreting this graph, this is a graph that did not force the treatment effect to be the same for every type of patient, but at the same time, it put a lot of restriction or Bayesian type borrowing across the covariate levels. And so we're not allowing wild estimates of the odds ratio in patient subgroups or specific patient covariate values. But our analysis that we presented earlier is indicating this should have been a single point. It should have been a spike here with no variation whatsoever. And so we're essentially estimating that the Bayesian variance of the random effects is 0. But we gave it the benefit of the doubt. And when you do that, you still see very little variation here around the central overall treatment effect. Now, if you were to plot the odds ratio that you estimate on a patient-specific basis against the risk ratio, you see tremendous variation in the risk ratio because risk ratios are not very good summary measures when you have heterogeneous outcome groups. And you see here that these very dark points have between 3,000 and 20,000 patients in them. So there's a lot of dominance of a few combinations here. And then when you look at the risk under the control treatment versus the absolute risk reduction, I think this is a very important graph. It's a very simple idea that if you trust your risk model and your risk model is not overfitted and it's pretty well calibrated, you can use your risk model to estimate the probability of death under the new treatment and the probability of death under control and subtract those two. So this is just the difference of two logistic models, and that's your estimate of the individual patient level absolute risk reductions. And we see that when you have the very low risk patients, and there's many of them, up to 3,000 patients here have, almost no risk, so they have no opportunity for the treatment to benefit them. You see this dramatic kind of variation when you compare the absolute risk reduction against the starting base risk. And so that's just a mathematical necessity, which we call risk magnification. And that's why we don't want to talk about heterogeneity of treatment effect, because this effect will always be there for any treatment. So absolute risk reduction would be a very boring metric for looking at HTE, because anytime the treatment has an average effect that's not 0, you'll see a relationship just like this. It has nothing to do with biology, medicine, pharmacology, or physiology. It's all math. And it's related to the fact that probabilities cannot go negative. They're tied down. This is the same sort of plot showing variation in risk ratio. So risk ratios are not very good for summarizing heterogeneous groups. But then this is the relationship between the risk and the control group and the odds ratio. So here again, we have, like one of these points is 1,600 patients. We have a very, very flat relationship between the risk under the control treatment and the odds ratio. And so remember, our best estimate is this should have been a single line. There should have been no variation at all. By giving it the benefit of the doubt, we see this little bit of variation vertically from 0.8 to 0.82. And we have a very small minority of patients with unusual covariate values for which we estimate the odds ratio might be a little bit more different from 0.81. But predominantly you see this flat relationship. And then I want to finish with this, this other blog article. This is by Laura Lazzaroni and myself. Laura's at Stanford. So this was called Electronic Health Records and RCT's Outcome Prediction versus Optimum Treatment Selection. So the main theme of this article is that electronic health records are not good for estimating treatment effectiveness, but they are good for estimating outcome tendencies. And when you combine outcome tendencies with the odds ratio or some other relative effect from a clinical trial, you can really have the best of both worlds. And so you end up with, there's a lot of intermediate material here, and there's a strong argument, I think, that very large electronic health records are not very good for looking at efficacy, and the size of the patient population doesn't overcome the bias from the EHR in any way. But I want to just leave you with this more general idea, which is from the Gusto 1 study. If you take what we did previously and you have your model that's a function of treatment, and you're assuming all the interactions are zero, like we believe they are close to zero, really, when you estimate the 30-day mortality risk for the control arm, that's just a plain old logistic regression model. And so once you know your age and kilo class, and there's a strong interaction between those two, so you need 4 age axes. Once you know your systolic blood pressure and it's truncated at 120, heart rate, previous infarction, location of infarction, you get so many points for all these and you add them up. That's on the log odds scale. And then you convert that to the nonlinear scale, which is your risk of dying. And then the difference between two logistic models is what's here. If you really did believe there's some interaction, you would need multiple axes for this thing. And you might have some very slight additional variation, but in our particular case, we did not think we needed to allow for that. And so the point here is when you have a way to estimate patient level risk, and so that you estimate patient level risk reduction, you're really taking into account risk magnification. None of this has anything to do whatsoever with heterogeneity of treatment effect, but it has everything to do with heterogeneity of patients. So I think it's very, very important to take into account heterogeneity of patients, and this kind of graphical device is just one way to do it. So I will close there and thank you for listening.
Great, thank you, Frank. That was a great talk. I think it really showed how graphical and data visualization techniques are really a part and piece of the analytical approach that we've been using towards looking at treatment effects and heterogeneity. The next speaker is Ilya Lipkovich. He received his doctorate in statistics from Virginia Tech. He's currently a senior research advisor at Eli Lilly and Company, and also a fellow of the American Statistical Association. He's published many articles, including those on subgroup identification. He is also a co-developer of subgroup identification methods and the co-author of 2 books. He's going to be talking about challenges of subgroup identification, analysis, and reporting, and Ilya will be sharing his slides. Thank you. So Ilya, you can start sharing your slides now.
Thank you for introduction and for inviting me. I hope you can see my slides now and hear me.
You need to share your screen.
I thought I did.
So far, I just see you. Can someone help? No, here they come.
Okay. Thank you.
Yeah, there they are.
So I will give a general overview of challenges of subgroup identification, analysis, and reporting. And I would like to acknowledge my colleagues, collaborators, Alex Dmitrienko, Bogdan Ratic, and David Swenson, representing the subgroup special interest group from SPI. And so I'll cover broadly several topics that of interest in this conference and being discussed actually yesterday and today. The guidelines on subgroup analysis, different phases of subgroup analysis in clinical data, and talk about principled or disciplined, as Steve Ruberg yesterday defined, data-driven subgroup analysis, methods for subgroup identification and analysis, and typical pitfalls in subgroup analysis strategies and challenges of graphical presentation of subgroups. And there are numerous guidelines on subgroup analysis over the years, and they stem from the fear of data dredging, as subgroup analysis often rightfully considered data dredging, and many authors came out with various checklists of principles for subgroup analysis. Just to mention a few, Brooks 2001 provided a list of 25 recommendations. Rothfeld 2005 proposed a guideline with 21 rules, and so on. And the general theme of these rules is pre-specification is important, biological plausibility, replicability, reproducibility, multiplicity control, not testing in subgroups unless spacing interaction test, or not testing in subgroups unless spacing the overall test for consistency, and so on. And the most recent EMA guideline actually came from somewhat different perspective. As on the one side, we have this subgroup analysis anxiety of the fear of sort of unsubstantiated claims of heterogeneity. But then on the other hand, we don't want the sponsor to ignore potential treatment effect heterogeneity. And so between these two sort of ends, there is a lot of recommendations and concerns. And for example, the draft guidelines advocated minimizing data-driven elements in subgroup investigation. In final version, they dropped that statement as somewhat confusing, I guess, because it's hard to understand how we can find subgroup where we don't do anything data-driven. So there is still vagueness about what exactly pre-specification means. As a presenter from yesterday rightfully mentioned that pre-specification itself is vague if it's not clear how it's combined with multiplicity adjustment, hypothesis testing. And pre-specification is one of those mythological words that used throughout many documents. For example, in draft guidelines, it was mentioned 14 times, and in the final version, 12 times. And so it's hard sometimes to understand what exactly elements should be pre-specified. So as we did a survey in 2015, we tried to understand what type of subgroup analysis actually exists, not to be sort of prescribed, but it was more a descriptive view of what actually people call subgroup analysis and how they do it. And we found in pharmaceutical industry, it typically could be confirmatory. On the one extreme, which everything is pre-planned, multiplicity is controlled in a strong way. And on the other extreme, this is biomarker subgroup discovery. However, both of these ends, as I would try to argue, can be principled or disciplined, as you can pre-specify subgroup discovery strategy with all elements. And so, of course, we have data-driven findings, and subgroups form dynamically, but however, you can control various elements. Whereas in the middle of this, you have somewhat loosely defined exploratory analysis and exploratory section of the protocols, or post hoc analysis, which naturally come after you find unanticipating subgroups effect, and you try to test for consistency, and you don't quite understand what's pre-specified, what's not. So speaking about principled or disciplined data-driven analysis, of course, it's important to pre-specify the strategy of analysis rather than what exactly candidate subgroups you would look at, because there is a variety of them depending on the scope. And what makes data-driven subgroup analysis principled is that complexity control to prevent data overfitting is introduced. Like in the previous talk, was this idea of penalizing treatment by covert interactions. And okay, what are the details? Of course, are important to know. How you define the penalty? Is it data-driven? Do you use cross-validation and so on? And so those elements needs to be defined in advance. The Taiwan error control of entire subgroup search strategy should be implemented, maybe perhaps in the form of false discovery rate or some other way, but you need to understand what's the Taiwan error of the findings, and you can do it Bayesian way or resampling under the null using frequentist approaches. And finally, once you identify some subgroups, you need to understand what's the honest estimates of treatment effect in the subgroups. If you do future study, what do you expect to find? And again, in this conference, we emphasize Bayesian methods, but again, frequentist methods could be used as well using resampling ideas. Now, what features of subgroup identification methods we should look for? We read papers, and there are tons of papers that introduce different methods. And first, you need to understand what is being done in the paper. What is the scope data that can be analyzed by the method? Is it just single variable, or is maybe 20 variables, 100 and 1,000? And what's the promise, and what's the model space defined by these biomarkers? As you can look at subgroups based on multiple biomarkers and only on single biomarker and so on. And what outputs does the method produce? It could be signature of promising subgroups, personalized treatment contrast, optimal treatment assignment for each patient, predictive biomarkers ordered by predictive strength. Is there a type 1 error, false discovery control? And is there estimate of, or honest estimate of treatment effect in those identified subgroups? And so recently we published a book chapter with Alex Dmitry and colleagues where we had this updated version of the table earlier published in Tutorial in Statistics in Medicine. Of course, I'm not going to go through this table, but just to give you a sheer idea, the idea of sheer magnitude of approaches that people developed. It's not just, of course, Bayesian versus frequencies. There are different elements that would define these different methods as, for example, what dimensionality, biomarker, selection type, control of false positive, complexity control, selection, bias control, how you compute honest estimate of treatment effects. And here we list the software in R typically available. So I hope this table would be useful. I am not claiming this is completely objective and unbiased towards our own methods, which were developed back in 2011. So what are typical pitfalls, challenges in subgroup analysis strategies? Identifying subgroups by multi-stage strategies, which is often the case. However, often involving human intervention. And then it's very hard to evaluate uncertainty associated with that because you can't replicate it and you can't program it and apply it to no data because there is different human interventions there that you cannot really code. And a common practice is to account for uncertainty of only the last step, forgetting about preliminary data logs that may lead to reduce number of candidate biomarkers or subgroups. And another one is it's often believed that no control of multiplicity is needed if analysis is done for internal decision making. And of course, if you don't control for anything, that's unadjusted analysis would inform wrong decisions, as often a sponsor would chase subgroups from one study to another. Now, adjusting for multiplicity after the search, which seems to be okay. You did a lot of search and then you do simple, perhaps resampling under the node or bootstrap or something like that, and you adjust treatment effect or compute the true sort of multiplicity adjusted p-value after the search. Typically, it shows that there is no subgroup and there is no heterogeneity. However, that because the procedure is greedy and found a lot of noise subgroups, which later are shrunk to zero. If you do this process as part of subgroup search, then using proper machine learning methods that maintaining bias variance trade-off, you would have better chance to find subgroups that would be replicable in the future studies. And finally, it's important to realize that no subgroup is a valid finding, so it doesn't have to necessarily present something. And so some people sponsor, especially fear that, okay, they don't find some interesting subgroup, the data is not useful. Now, we also maintain a page where put different methods and software, so some people actually use it. And if you send me your tool, if you develop a tool, I can put it there. Now, going to challenges of graphical presentation of subgroups. Well, one big challenge is that if you do a lot of search, or maybe not a lot of search, but some search, and you do data-driven candidate subgroups, then you present it, then how you account for uncertainty associated with that. Because very commonly people present graphical presentation of subgroup. They don't reflect any selection that could have happened before that. As an extreme case, there is a very interesting paper that was an eye-opener for many people, I guess, from Richard Simon and colleagues using cross-validation to evaluate predictive accuracy of survival risk based on high dimensional data. So there are really a lot of variables there. And this graph I borrowed from this paper, where on the left you see one simulated data set, when you see survival curves for low and high risk that were defined by using the machine learning based classifier. And you see that, okay, you found some subgroup of patients with really good low risk, good survival on the top. But now if you do cross-validation in that same replicated data set, there's nothing, there's no difference. Difference is shrunk to 0. And so that's something that is often important to understand. Now, but okay, there is more benign, seemingly more benign case. Okay, you don't really seem to do any selection. You just present a forest plot or your favorite plot, for example, upset plot, which were advertised in recent interesting review paper by Ballerini and colleagues. And here you don't really hide anything. You show all subgroups and even all the combinations of them, which is very nicely shown here. As you see, 3 binary biomarkers, and then you can plot all possible subgroups you can form from them. And here, these 3 columns indicate which subgroups are on or off and which variables are not used. So you can see all possible subgroups, and the idea is that people would integrate multiplicity in their head. But how the plot will be used? It will be used for decision making. So somebody will say, okay, we're going next, our trial will be in this subgroup of these patients with these 2 variables, high level and this low. But or maybe you just report it somewhere and people would use it for meta-analysis later on, and then they want to know what's the effect before adjustment. So there is always uncertainty about how you actually report the subgroups so that they don't induce false positive conclusions. And so one approach proposed recently by Dennis, standardized effect adjusted for multiple overlapping subgroups, where you see essentially the same data that you would plot on the forest plot, and they are ordered here by standardized differences. However, by permutation, you obtain the distribution of the standardized difference using all these variables, sort of permuted treatment under the null, or permuted, or maybe using more complex permutation schemes. And here you can have idea which subgroups are really beyond those adjusted 90% probability interval. And of course, it's much wider than you would just expect without adjustment. And finally, I'll present a simple example which illustrates how the data scope is important to anticipate what effect you would get if you replicate your trial in the future. So there is 470 subjects here, and there is 317 treated, 153 control. And outcome variables binary mortality. So it's sepsis study where a lot of people die after 28 years. And so the mortality rates were about the same for treated and control, with one-sided log-rank p-value 0.83. So there seems no difference. Now I planned, there's 8 biomarkers, mostly the numerical variables. And so I planted myself a subgroup which shows that there's a large difference between treated and untreated in patients for whom time to the first organ failure was relatively short and age was larger. And so there's like all variables here. And so I use this to plant a subgroup. Just because I planted subgroup in this one data set doesn't mean that you would find this in the future data, of course. So and that brings a question, what's real and what's not. So of course, so the difference, apparent difference is about 28% between treated and control in this subgroup. And so this plots this observed difference and confidence interval. And so it seems very promising. The lower confidence bound is way below 0. Now, what if you use random forest? Using generalized random forest, you can compute out-of-back prediction for treatment effect in this same subgroup. But now you allow yourself to, of course, different cutoffs. So you have 2 continuous variables, this time to the first organ failure and age. You feed it in this generalized random forest, and you see that the effect shrunk down to 0. And so it's not Bayesian, it's frequential. So in this conference, there's more interest, of course, in Bayesian shrinkage, but you can do it also using frequentist methods for estimating causal difference. And so you see with 2 variables, it shrank down to 0, but it still looks promising. Now, if you add 3 more variables beyond time to the first organ failure and age, then it shrunk down even more, and now the bootstrap-based confidence interval doesn't look very good. And then you keep adding variables, you add 9 more variables, and then you pretty much, you still get some effect, but it's very questionable. And so now when you do a future trial, probably you shouldn't expect that the effect would be as observed. And so now when you plot it, then you do you need to adjust for those other variables using Bayesian and frequentist methods or not? That's something that people obviously would like to know. And to summarize, subgroup analysis in clinical trials remain a challenge, attracting numerous guidelines. The difference between pre-specified and data-driven subgroups is blurred. Data-driven elements are often hidden, so there is hidden multiplicity, and not everything may be pre-specified, and there is human intervention in the process, which is always often hidden. A wealth of methods for subgroup identification emerged in the last 10 years, contributed from multiple fields, including causal inference in machine learning and multiplicity. Graphical presentation of subgroups is often disconnected from the methods of their construction and may lead to exaggerated view of treatment effects and incorrect decisions. And that's, I have some references, so thank you very much.
Great. Thank you very much, Ilya. That was a great talk. I think it really illustrated one of the underlying difficulties in understanding heterogeneity of treatment effects, which is how, in fact, we define subgroups. There are so many ways to define subgroups and no clear agreement on that. And if that's really the foundational first question to looking at heterogeneity, it just makes it even more complicated, not only to define the subgroups, but once you define them, then how you handle them. So again, thank you very much. It's very provocative and gives us a lot to think about. Now I'd like to introduce the last speaker, Michael Griswold. He's a professor of biostatistics and data sciences and directs the Memory Impairment and Neurodegenerative Dementia Center Scientific Evidence and Technology Core. That's called Mindset. And has also an adjunct appointment at Johns Hopkins University. He founded the UMMC Department of Data Sciences, and he's the PI of numerous contracts and grants. He's directed biostatistics and data science centers at many institutions, and he's going to talk about interactive visualizations. Thank you, Michael.
Thank you, Marcia. Can everyone see my screen okay?
Yes.
Lovely. Thank you for the invite to talk at this wonderful conference. I'm going to talk about interactive visualization for HTE. Sitting around my slides, so I've got a couple of caveats, more than disclaimers, but a few shout outs to my colleagues at the Annals of Internal Medicine who always taught me how will this research change clinical practice. My wonderful Mind Center team here, Chad and Jim and Rajesh, without whom there's no way this is possible. And to the our visualization session with Frank and Ilya and Marcia and Barbara. We met, of course, as many of you did, a month ago, and Barbara issued us these 3 challenges. How do we extrapolate from an RCT? How do we appropriately help people interpret any potential HTEs? And how do we convince people that these results speak to them? So I'm going to try my best. We've worked for the last 3 weeks and trying to put this together to show you guys some stuff. I'm going to start with an example, a fun one called The Voting Habits of Americans Like You from the New York Times, which does lovely interactive visualizations. This one's from 2012, so not from the very latest election, thank God. And it shows initially in 2012, overall as a country, that 60% of the people voted, and that's on the y-axis here, the turnout. And about 51% of them voted for Democrats, and that's on the x-axis, the Democratic voting. And then they say, okay, let's see, our primary message that we'd like to do is something about racial divisions. So they're going to color scheme it by 4 different categories that they defined. And they show that in 2012, whites and blacks were about equally likely to vote, 62% versus 61%. But a majority of the whites voted for Republicans, 41% voted for Democrats, and a majority of blacks voted for Democrats, 95%. But then we could split that out and see what education effects there were. So splitting by education, the circles kind of split vertically. People who are higher educated tend to vote more. People who are less educated tend to vote less. And also on the x-axis, people with the highest education and the least education tended to vote more democratically, giving rise to these C-shaped curves. But then we could split it by sex. Sex, in contrast to education, is a horizontal split. women tend to vote more democratically than men do within each of the other subgroups already defined. And continuing, how about age? Age actually was a vertical split. It affects voter turnout. So you have higher voting percentages at older ages. And then what about by state? Good Lord. Now there's a variable with 50 at least categories, and you can see that it basically fills the entire plot now. And they have a nice journalistic interpretation that there's at least as much heterogeneity amongst the whites as there is between what are more commonly known as the racial divides. And then they can have a temporal aspect where they show in 2004, this is what the plot looked like, and then up to 2012, which they interpret as the polarization of America, unfortunately, as we kind of get more farther apart from each other. And then at the end, they have this lovely find yourself. How does this chart speak to you? And if you click on that, here is the visualization. And you could say, well, let's see, for people like me, I am a white male who is, let's go with 30 to 44, those are good years. I am overeducated, and as I'm clicking through here, it is drilling down deeper and deeper into the data. And I was born and raised in Anchorage, Alaska, which was 75% voted and 41% Democratic. But then I did my undergrad in Minnesota, and they were voting higher rates and at higher Democratic rates. And then I went for my graduate work to Maryland in Baltimore, where I met Tom and Ravi and other wonderful people. And this is about similar with Minnesota. And then at grad school, I met my lovely wife, who's from Mississippi. And so now I'm in Mississippi, which looks a little bit different. And if I wanted to say, well, where else might I like to live in the country for people who look kind of like me, I could take some of these guys off and say, well, there's all of the states for middle-aged, let's say, overeducated people. And I might say, let's compare that to people who don't have as much, didn't have as many blessings that I did for their educational perspectives in life. And there's large disparities there. So coming back to my talk, I hope that was kind of an interesting little, little exercise. We used to, in interactive visualizations, we would just produce the visualization and say, okay, I hope people go and click on all these little buttons I made and come up with a story I'd like them to find. And it didn't work very well. So what has been done over the last few years is the development of kind of journalistic interactive visualizations where these interactives are partnered with what is, I didn't make this up, scrolly telling, where you lead your audience through the data and your message, helping people navigate the complex information and what the graphic is. In essence, teaching them how to interpret what you're trying to tell them about. So fantastic conference. Agree with everything that everybody has said so far about the methodologic challenges, 200 years of which. I really like the Stephen Sen's article. I think it's very readable, and even for lay people. My wife loved also this graphic that was in his article. Are we ever going to get over to this little island? There's a lot of crazy stuff happening around. But you play that against a great talk by David Kent, where the multiplicity of attributes that a clinician has to think about when they're trying to think of how to care for their patient. And did a computer amalgamation of all these individuals and came up with the average person, who looks kind of like everybody else, but doesn't really represent any one of them very well. So there's a tension. There's a methodologic versus a patient care tension that inherently occurs, and it's been talked about very well over the last couple of days. How can visualizations help is where I'm going to try to land today. If we're going to look at a visualization, we need a method to implement. I'm going to use the PATH method, which just came out. I was blessed to be an editor on this. It was put together by just a steer group of people. I think some of you are on this. Ravi, I think you're on this. They developed consensus guidance for what they called risk modeling approaches versus effect modeling approaches, a lot of which, again, has been talked about here today. Just a very tiny bit of math, Barbara, which is basically a clinician thinks about like this, all these different factors that are I'm staring at with my patient. How do I build those into my mind with some kind of a risk model that says how likely they are to get the event, how likely they are to get the outcome? And the PATH approach was to say, it was an aggregation method to say, well, okay, we'll use that one variable, that overall predicted risk in your modeling, instead of all of these individual factors all together and simultaneously. And then if you want to look at an HTE, well, now you've got one thing to look at, one interaction term, in reducing the multiplicity issues. And you can say, how does the treatment affect people at higher or lower levels of risk? And that's instead of or versus to an effect modification approach where you would have all these different P interaction terms that are needing to be shrunk and smooth and all those kind of good things we've talked about so far. A reduction in multiple testing and a focus on clinical practices where these guys hit. And we also have some data to apply a method to. So I'm going to use the SPRINT trial, which all of you guys probably are very well aware of. Arm 1 was standard treatment down to 140, intensive treatment all the way down to 120. Primary outcomes, CVD. They had some important exclusions. Diabetes. I'm in Mississippi. There's a lot of people who have diabetes down here. Subgroups available. They a priori biologically specified, as Ilya was just talking about, 6 different things to look at. And they said, we think that these are important to look at, but we're not going to include them in any kind of actual testing paradigm because of all the issues we've already talked about. So they produced the standard force plot over here that thankfully all lined up on the correct side of the bar. But that did not stop another 50 to 100 papers being written on subgroup analyses that we all just talked about, comparing, well, what if we split it 75? Well, what if we split it at 80? I'm waiting for the 85 and the 90-year-old papers to come out. The gender, race, ethnicity, CKD groups. The most fun one I've seen so far is this combination paper where they looked at and said that the greatest impact is younger patients without CKD or CVD, or in older patients with CKD or CVD, which is just all the fun stuff we've been talking about here today. So we took all that and we wrapped it up and we said, hey, let's try to think about doing something like the New York Times did. So here is intensive versus standard blood pressure treatment in people like you. And you can link out to different information about SPRINT and other things like that. In the SPRINT trial, here's your 9,000 participants, and they're about 6% chance just on their baseline factors of them having one of these CVD outcomes. If you split it by our primary story, treatment, you have standard and intensive, the 6.8 versus 5.2% probabilities of CVD event are the trial's primary outcomes. And that's what they came out with in their paper. We could split that by age because that was something that they thought they wanted to look at. And you can see here that, yeah, older people, they have higher risks of CVD events. Nothing new there. You could split it by sex. And now you can see, okay, well, again, there's that horizontal split. Unfortunately, guys, we're usually at higher risk for stuff. 12.4 versus 8.4% on standard versus intensive treatment. The older men being the highest group. You could split it by race. And you can see, okay, well, now I've got like old white men up here, and I've got young Hispanic women down here. But instead of kind of focusing on those things, you can use these storytelling and journalistic features to tell a little bit different stories. Like, you know what? Sure, you could say that, but the size of these circles is related to how much information there is about the group. And results in smaller groups can be, stealing Tom Lewis's phrase here, highly unstable. And in fact, all of their different categories that they looked at, you could split it out and you could say, hey, check this out. Look at this scatter shot of teeny tiny little dots. All of those guys together actually comprise the overall study results. The dirty little secret of statistics, it's all just weighted averages. So these are the underlying components that get weighted up, whether you're using Bayesian or frequentist or likelihood technologies, into the overall results. And you can say none of these specific subgroups are large enough to determine whether the treatment worked better for them than one, right? But in general, you do see a pattern of kind of lower rates overall in the intensive, which in the Stevenson sense says, well, keep that overall consideration in mind. We can embed overall averages or group averages within that visualization so people can look at how it speaks to them particularly. But when there's, as often, not enough information, then you rely more heavily on the overall trial results. And you can link out to things like, well, here's a personalized care Nature article that Stevenson referenced in his article. That's the only reason I put it there, Steven. If there's a different one that's better, no problem. Here's a link out to go read this. Go look at the big octopus and see what all the issues are. And then go find yourself in these charts. Can you find yourself? Sure, you can find yourself. Here's that same chart now that we built the story for the patient and the clinician and say, here's their baseline outcome risk. Well, my wife, who is a geriatrician epidemiologist, treats older women and men. But if we said that she's staring at an older woman who is, let's say, African-American, and here I will go down a little bit so we can see as we go, and didn't smoke but is overweight, had CKD, let's say, but there was no pre-existing CVD. Well, that is the person that she's staring at right in front of her to try to treat. But there were only 13 people like that in the study. And there's only 7 people on standard treatment in that study. So you put those together with the overall treatment results, the 5.2% and the 6.8%, and you have a conversation about that. And if you wanted to, you could say, okay, well, let's put on, for context, another one, like everybody who is older. And there's the overall marginalized estimates for people who are older on standard or on intensive treatment. Or you could do it for ethnicity of interest. And you can keep building your stories. And maybe you're not just interested in the CVD event, but maybe you want to tell that kind of risk benefit if they had any serious adverse events. Okay, well, then they had a lot of adverse events in there. There wasn't a huge difference overall, as you can see by the overall ones. And again, for the specific subgroup, there's not a lot of information for me to tell one way or the other. Injurious falls is another one. If you're about to put a older adult woman on a third hypertensive medication, then one thing you worry about is, are they going to fall over and have a deleterious effects from falls? So then you get to see all that same information now for discussion in context with whatever individual results we do have. If there's enough there, great. If there's not, then here's what the overall marginalized look like. And then you could say, well, all right, let's if we reset these guys. Another important part for visualizations is being able to reset quickly. What if I was a regulator instead of a clinician or a patient? And I wanted to say, well, what if there was no heterogeneity of treatment effect? What would that look like? Well, that is this model down here. That says that there's no difference on the log odds scale. And from what Frank just talked about earlier, as you see, the higher the baseline outcome risk on the absolute scale, the bigger the risk difference, just mathematically, if you're modeling on log odds scales with linear assumptions. Then if I slide all the way here, here's all these different models that the path group came up with. And you can see how those individual estimates are changing with the different pooling, the different shrinkage that we're applying until we get up to all the way up to this totally non-parametrically, just use the individual group information. And it would be kind of tempting to hover over this one and say, well, great. I should put all of the 75-year-old men who are white and former smokers with normal weight and CKD and CVD on treatment because none of them had the event. But clearly there's only 14 people in there and there's not enough information to be able to say anything about that specific result. And you've taught them that through the scrollytelling. So back to some slides. Interactives, great for teaching a viewer the concepts and limitations. Great for engaging people and showing them this speaks to you. Very nice for finders and keeping things in context about what the averages are. And I'll tell you a little bit more on the averages in a second. For regulators, examining sensitivity assumptions. Most of the stuff we've talked about at this conference are Bayesian ideas, fantastic ideas, probably better than what I've shown here for how you borrow information across different groups of people and where my assumptions are can often be represented by some kind of tuning parameter. The rest, these next couple of slides are math. I'm going to skip them for time and for Barbara's mental health. But generally, this is how we mapped the path equations. They laid out these different equations. You can map these, as most statistical techniques, into some kind of overarching framework where all of these are based on some kind of slider. That's how we slid around to those different assumptions. And I forgot to tell you, I forgot to show this, if you click on that middle bar, it brings up all of these equations and the framework underlying them. But we slid these down just by modifying then the sliders. That's the same idea that we've talked about for, Frank just talked about penalized regression. Elastic nets are defined, they're a generalization of support vector models and lassos, and they are defined by a couple of smoothing parameters and shrinkage parameters. Bayesian hierarchical models and regularized neural networks and machine learning, deep learning AI tools, they are mostly defined or can be thought of as having these tuners, these sliders that take you all the way from a complete non-HTE world to seeing all the way through to where the individual estimates themselves non-parametrically would rely. And then you can say, well, here under AIC or under pick your metric of interest, root mean squared error, loss functions, here is where I think the best lambda parameter is, but I'll still let somebody slide that around to see how sensitive, how robust are those answers to choices of priors or borrowing strength. I talked about marginalized as a quick note. There's a difference between marginal and marginalized. When folks on day one were saying these marginal estimates are bad, what they were saying was if you take just all of the older people and you do it for them. But that, again, there may be big covariate differences between older people and younger people. So instead, we used what Ravi Baradan talked about yesterday, a marginalized group averages for all of our scrollytellers and our group estimates. You take your richest model and then you weight up in order to obtain unconfounded average group estimates. And both of these techniques, the sliders and these marginalizations, they can be done independently of your actual estimation technique. you give me a set of estimates for all of the individual subgroups, and you give me the settings, just like Frank just showed. No, I'm sorry, it was, yeah, it was Frank, I think. I'm sorry, one of our previous Bayes, no, it's Don Barry, I'm sorry. Don had 15 slides that he had somebody click through to show how the change in the tuning parameter changed the different estimates. That's great, that's the exact idea that we'd want to say, just give us those estimates, we can push them into this visualizer, and you could slide it around instead of having to click on 15 different slides. Lastly, expansions. I showed probability scales because that's how I've been taught clinicians think, not even just in terms of absolute difference, because a 1% difference means something different if it's a 1% versus 2% or a 51% versus 52%. So I generally start with just the actual risks. And then I say, well, great, we can add quickly little tabs down here for show me different scales, show me the risks, show me the absolute differences, show me the relative risks, show me the log odds ratios, which is what I usually model on because they're just better canonical link functions. You could add different extensions for grade. Here's another connected plot to the original visualization that could use upset and step and other frames similar to what Ilya just showed. And importantly, we didn't put this in yet, but you could show the diagnostic of where the AIC sat depending on where your slider was. Stuart Pocock had a fantastic talk. I loved it. And I actually asked him if I could borrow this slide because his picture was much better than mine for demonstrating what a clinician actually thinks about is risk-benefit trade-offs. And the benefits often have to be much higher than the risks for them to actually treat the patient. So imagine this risk-benefit as being another 2 sets of clickable items that you could say, great, I don't want to just see the baseline outcome risk and the risk here. Show me what the benefit risk trade-offs are, but let me go find which one of these little dots and make the dots the size of the group that I'm looking at. Actually, I'm talking to right now. Who is that person? And along down the line, more and more and more things. The last thing I'm going to say is interactive extensions. So another dimension is adding data. You can collect data through interactives. The Tilda study looked at the Sprint study and said, those people don't look like our people all that much, I don't think, because we get a lot more of the adverse events than Sprint reported. That's kind of interesting. Well, great. Wouldn't it have been wonderful to say, hey, researcher, click on this button, format your data in this way with these column headers, and embed your data into our visualization so you could actually better directly compare what's going on instead of just these bar charts. Extend that to an individual and a clinician in an N-of-1 trial where they could put in, great, these are my characteristics. I've made a treatment decision. I'm going to put them on standard. I'm going to put them on intensive. Great, come back 2 months later and put in what your outcomes were and build the data that has been treated. You could separate that as a different category in the visualization, but also let it look at pulling into the overall results. Another hat I wear is for data harmonization and data commons and building data ecosystems. A lot of those, like the NCI's portal and the NHLBI's new biodata catalyst, they are built because they have to be by computer scientists and data scientists who are fantastic at huge data issues and computational algorithms. But what you get out of those are kind of bar charts and pie charts if you're lucky. They're not being built to very efficiently get out interactive visualizations. You can't have a visualization talk without a picture of Edward Tufte. He always said, start with the end in mind. Think about how you want to present and how you want to communicate the results to whoever you're talking about. And then build your research, build your data, and build your analyses. So I would encourage all of the prestigious people, much more than me, on this conference to go talk to your local computer scientist, your data commons architecturalist who are out there right now, and try to get them to know what these perspectives are. Because if we try to jury rig it after these are built, it's going to be much tougher, much tougher. So I really thank you. A couple other people already had Richard Pito quotes. Always do them, but never really believe them. Put that into the context of what the overall trial said. There's been 200 years of multiple methodologic and clinical challenges. I hope that some of these new technology ways are ways that we can help start to overcome some of these things. The path approach and the find yourself analysis, specific ones that we created, we did these in the last 3 weeks because Barbara gave us these challenges. We want to try to meet them. They are meant for examples. I'm not recommending us to go with a path approach. I like the Bayesian ideas actually much better. And those sliders are inherently involved in the Bayesian ones as well. You can maybe help make all the difference by getting yourselves in with your local data architecturalists. For poetry buffs out there, I snuck in some Robert Frost quotes throughout. you can go back and check those out. It's not two roads diverge. We should all be together, coming together to find ourselves on good paths. Thank you very much, and I appreciate any feedback after the talk later on.
Michael, thank you very, very much. That was a great approach and different view on how we can actually take this data and make it more usable and interpretable, make the subgroups usable and interpretable to patients and physicians. I think it ties in very nicely with Frank's terminology, differential treatment effects rather than heterogeneity of treatment effects. It shows a spectrum of effects. And I think tying this to Bayesian and other kinds of analyses would be a great contribution to this area. And I'm going to, we've used up our time, so I'm going to turn this over to the next panel, which is a discussion of statistical methods. Thank you.
Hello. First, I'd like to thank all the speakers and presenters from the previous sessions and panels. I thought this has been a wonderful meeting, and I really enjoyed all the presentations. This next panel, well, first of all, I'll let you know who I am. My name is Thomas Gwise. I'm the director of the Division of Biometrics 9 at CEDAR at FDA. Our division reviews products for hematology and oncology. So I'll be moderating this panel session, Statistical Methods, Pros and Cons. In this session, through a series of questions, our panel will try to bring together major points discussed over the last 2 days. I'll now introduce our panel members. Qi Jiang is a vice president and head of biometrics at Seagen. Before Seagen, she worked at Harvard, Merck, Novartis, and Amgen, accruing many years of diverse experience in drug research, development, global submissions and approvals, and commercialization. She is an ASA fellow, has over 90 peer-reviewed publications, is co-editor of 2 books on safety and benefit risk. Currently, she is co-leading the ASA Biopharm Statistical Methods in Oncology Scientific Working Group. Frank Rockwald is a professor of biostatistics and bioinformatics at Duke University Medical Center, affiliate professor of biostatistics at Virginia Commonwealth University, and managing partner of Rockwald Incorporated. His career includes senior management positions in industry, most recently as Chief Safety Officer for GSK. Dr. Rockwald served as Chairman of CDISC and President of the Society for Clinical Trials. He also served on the initial PCORI Advisory Board and is currently on the boards of the Frontier Science Foundation and an advisor to EMA. He is an elected fellow of both the American Statistical Association and the Society for Clinical Trials and the Royal Statistical Society. John Scott is director of the Division of Biostatistics and FDA Center for Biologics Evaluation and Research. Prior to joining the FDA in 2008, he worked in psychiatric clinical trials at the University of Pittsburgh Medical Center. Dr. Scott holds a PhD in biostatistics from the University of Pittsburgh, an MA in mathematics from Washington University in St. Louis, and a BA in liberal arts from Sarah Lawrence College. He's a fellow of the American Statistical Association and is currently an editor of the journal Pharmaceutical Statistics. Can we go to the next slide, please? And the next slide. Over the last 2 days, we've heard some common sweeping generalities such as, always do subgroup analysis, but never believe them. Be cautious about over-interpreting results. Be cautious about inferring differences, etc. I think we're all looking for situations where these broad generalities don't hold completely. Please discuss how some of the methods showcased over the last 2 days may help allay our concerns, or why the cautions should remain strong. And if one of the panel members would like to volunteer and go ahead and take a stab at addressing this question.
So maybe I can be the first for this
thank
question.
You, Chi.
Sure. Thank you, Tom, first of all, for your nice intro. We very much appreciate. And also, I like to thank meeting organizer and for the great opportunity being here to share our thoughts with you. And this is such a fantastic conference with numerous excellent presentation. And as we know, subgroup analysis topic is very important, but remains very challenging in the field. So there's quite a few concerns related to subgroup analysis. Just to name a few, like very small size, there's a very low powers there, there's many of them, and pre-specified and versus ad hoc, etc. So I do not think there's a one single methodology can be utilized in the drug development, which can address all of those issues at the same time. But nevertheless, to me, is continuous true is there's quite a bit of the methodology and the thinking need to be put together as a totality when we thinking about, the question, Tom, you put there, and to start from design stage, right? So over my long and the short journey, whichever words you're putting there, and I noticed that people often put a very long list of subgroups there. So, like there's a 15, 20 of them, then analyze each of them for each of the primary, key secondary, and sometimes even many other endpoints. So as a statistician, we know, and there's a false positive rate there, and we may just by chance alone find some odd results over there. So the efforts start from design stage is we really should put those subgroups which are prognostic factor. And also if some subgroups, when, for example, when you look at the two levels, one level it only has a 5% of patients, the other level has a 95% patients. So very often you will run into the small sample size issue. So there's a tons of things we need to watch out during the study design stage at the beginning. Then we have a study conduct. We get the data, we analyze those, and we need to report them and interpret those. And there's a lot of the methodology, and many of them presented by our wonderful speakers today. And I continue to think, the basic methodology plus those more innovative methodology presented, they all useful as a totality of the thinking there, starting from point estimate. Of course, point estimate for each of the subgroups still need to be there. And the confidence interval does provide us the information on the uncertainties, right? So should be presented over there as well. And we also talk about interaction testings. And yes, the power is very low, but it's also gave us some information there. Then on top of that, the graphical presentation, I think just a few minutes ago, Michael gave a very nice presentation. He not only gave his own presentation, he summarized the great points by all those excellent presenters before him, right? So start from basic, the forest plot, and it's very straightforward, and many people who doesn't have a lot of statistics background can understand them very well, but with the caveats there. Excuse me. So there's a modified forest plot and plus all kinds of other and the plots and the talk about the benefit to risk. So bottom line, the graphical presentation can be very helpful there. And one more thing is, yes, shrinkage estimate. I know I saw those chat and the Q&A, and we tend to say Bayesian shrinkage estimate. I know some of the speakers' audience felt it's not necessarily Bayesian, but regardless, the shrinkage estimate could be very helpful. Then we can borrow the information from the relevant groups over there. So I think at the end of the day, summarizes all those techniques are very helpful, regardless more basic one or more innovative one. And we do need to look into all of those as a totality of the consideration. On top of that, of course, we need to consider biological possibilities there. That's always the part of the default thinking there. So I just pause here and let other panelists share your thoughts. Thank you.
Okay, I'll take the next crack, John. Thank you, Chi. I'm not going to repeat. I agree with a lot of things that Chi said. The first thing I want to do, though, is express my appreciation to Barbara and Steve Snappin and Charday and all the FDA colleagues and everybody else who participated. It's been a pleasure working with you over the past year and a half setting up this meeting. And I echo Frank Harrell's comment. My expectations were really high, and that the presentations and the information presented here succeeded that. I think there has been some great discussion both yesterday and in some of the talks yesterday about what are we looking for? Are we trying to confirm the overall treatment effect and show that it's generalizable? Are we looking for a particular subgroup in terms of diversity? I think the question is, I think there's some belief that if you're looking at the regional examples like Adon Berry talked about, there's belief that you're going to have some regional differences in healthcare in a trial. So it's not unanticipated you're going to have some heterogeneity. So the question is, what are you looking for? What I liked about yesterday, I think Scott Evans and Stuart Pocock did a nice job, and Michael Griswold highlighted that today, of integrating benefit and risk into the equation. Because we spend a lot of time, a lot of the methodologies are trying to look at the overall treatment effect, primarily for efficacy. And I think integrating benefit and risk is an important piece here, particularly as Michael highlighted in his graphics. One thing that was not discussed a lot here, and it makes sense based on the way we set the meeting up, but I feel important to mention, when you're looking at heterogeneous treatment effect or looking at subgroups, most of the approaches today assume there was some overall treatment effect in the trial, and you're trying to understand whether or not that applies across all groups, or if there's some group where maybe the effect is less optimal. We didn't spend much time talking about subgroup analyses in trials where there is no overall treatment effect, which I think is where the cautions would be, in my opinion, would have to be much greater. And if you follow the Biogen Advisory Committee meeting of a few weeks ago, I think that's worthwhile to look into. That's what was based around subgroup analyses. I repeat Steve's comment yesterday. I'm not a Bayesian by practice nor expertise, but the Bayesian methodology here just seems to make sense. People have prior beliefs and prior understanding. And so I think that approach, and I think I agree with, that was really educational, Tom and Don's presentation today about sort of the robustness of the shrinkage estimator and the exchangeability assumption. That was extremely helpful to me. I also want to echo a comment that was made, I think, by a couple of speakers yesterday, that, and I think Frank Harrell emphasized that today, that looking at this as a model-based approach as opposed to picking out dichotomized subgroups, I think is an important message to send to the population, because I think that's where, I think Michael did a nice job in his visualization today. Lastly, I do want to, I think Stuart's comment yesterday, which I like the way he characterized heterogeneous treatment effect. If you can either have a quantitative or qualitative effect, which is the commonly used terminology, but I like his all or none statement, because I think that is an area where people have got, in terms of the regional effect, has been stated well. So I'm going to stop there. Again, I agree with a lot of things that she said.
Thank you, Frank.
Okay,
John?
Sure, yeah. I'm now in the enviable position of being able to agree with a lot of what both Frank and Chi said. Starting with the thanks to Barbara and the other organizers. It really has been an excellent first couple days. On the question as stated, I guess I would land more on the cautions should remain strong side than on a straw man, go ahead and infer differences side. Outside of a few distinct areas, I think subgroup analyses are rarely quite reliable and mostly should be considered as candidate hypotheses for further study. If we're going to sort of parcel out specific areas where this is less true, certainly if you're looking at biomarker-defined subgroups for targeted therapies, that's somewhere where we really would expect heterogeneity. In some cases, a very rare situation is a truly confirmatory subgroup analysis where something's been found in phase 3 and there's another phase 3 to confirm it. And I guess echoing what Frank said, I would reserve some thoughts based in this area for very large effects, especially very large regional effects in large trials in the presence of different backgrounds of care. That's a situation where I wouldn't say we can proceed confidently, but at least we might really be working with something there. But going back to caution, one of the things I wanted to mention that kept striking me over the past couple days, in most of the examples that people presented where there were subgroup findings that are still currently believed to be true in one way or another, most of those came from very large trials, especially very large cardiovascular trials. And it's quite obvious, but I'm not sure I heard anyone say it specifically. There's a lot less we can learn about HTE in a few hundred patient oncology trial than in a 10,000 patient CV trial. Again, very obvious, but very important to keep in mind. And a lot of the time at CBER, we spend time with some very rare disease trials with fewer than 100 patients, sometimes many fewer than 100 patients. And I think it's really only a very mild exaggeration to say we can't learn anything at all about subgroups or HTE from those trials. So just sort of managing expectations and keeping in mind the limits of what can be known is important.
Thank you, Scott. May we have the next slide, please? Thank you. We heard presentations describing several motivators for subgroup analyses. Among these are informing prescriptions, or maybe confirmatory would be another word for this. Looking for biomarkers of heterogeneity, safety signals, and some others post hoc was mentioned as a listing. Please share your opinion of specific approaches that may be more appropriate than others in certain situations. And since John was last for the last question, I'll ask him to go first and give a fresh stab at this.
Sure, I'll be happy to, Tom, thanks. So one thing I like about this question, although sub-bullets, it really emphasizes how many very different things we're talking about with subgroup analysis and with HTE. And Ilya's talk in the last session also brought this out quite a lot. Just to express that a little provocatively, it's not really obvious to me that talking about demographic specific prescription guidelines on the one hand and biomarker discovery on the other are actually the same thing, or should fall into the scope of the same workshop or discussion. But to generalize as best I can in any of those areas, I think it is useful to think of these analyses as decision support. What decisions are going to be made based on the results of the analysis? Whose decision is it? Is it a regulator's decision, a scientific decision for further study, or a decision made by a prescriber-patient dyad? And what information is needed to make that decision? One thing I'll agree with that Tom Lewis said this morning, I think hypothesis tests have really essentially no role to play in HTE evaluation. There's maybe some very few exceptions, like a confirmatory trial following up on an observed qualitative interaction, but that's rare. And for the most part, what we're looking at are instead discussions of subgroup estimates, estimates of uncertainty, and displays. And we've seen a lot of different flavors of shrinkage estimation among the presentations in the last couple days. And I I wouldn't commit to one being better than another for a specific purpose. I think they probably all have roles. I also don't think it's necessarily a bad idea to look at a spectrum of approaches in a lot of cases, because I think the really critical thing we want to do is to accurately convey uncertainty, and showing an entire spectrum of different assumptions about tau, as Don Barry had, and different approaches entirely may be as good a way as any to try to communicate not only our best guess for subgroup estimates, but also how little we may know about it from one trial. I'll turn it over to Chi or Frank. Thanks.
Hi, Frank, would you mind going next?
Sure. Start off with one of my first comments was going to be, which now is echoing John's comment. I was very taken by Tom's comment as well about the role of testing in this process. I'm not sure that's really the primary direction we should take. And one of the things I was going to focus on here in this question, I spent a lot of my career focusing on safety signals, and I learned a lot actually in the last 2 days about the application of these methods and looking for safety signals and differential safety signals. And I think Scott and Stuart yesterday, just to repeat what I said in the last question, focusing on benefits or risk really brought this attention to that. My only caution in the safety world is often safety signals are based on a combination of events and a combination of a thought process. So I guess more I studied and learned today about shrinkage estimators, I would want to make sure caution against blunting a signal by borrowing information that actually downplays a signal that could be on, say, 3 or 4 parallel tracks of different events. But other than that, I think it's a useful tool. The other thing that I think is extremely important, particularly around safety, and if you look at benefit to risk, is safety is entirely, to me, belongs in a Bayesian mindset. You don't ever, particularly if you're the clinician looking at a safety signal, everything is based on prior belief, either due to the class or animal data or something you learned in phase 1 or some other piece of information. So I think bringing that into the way to estimate looking at different subgroups is actually essential. And I personally learned a lot today by listening to Tom and Don and some of the other speakers about how to utilize this in a safety perspective. And I know that wasn't the primary driver of a lot of the discussions, but I certainly would encourage more work in that area.
Thank you, Frank. Chi?
Sure. So this time I'm the last. So I absolutely, agree, and I like to echo one of the many thoughts John shared, and looking into the spectrum of the methodology. To me, is whatever we do here, the very often so we should clarify what are the clinical questions, what are the objectives. Then based on the clinical question, based on the objective, then we come up with the methodology. But because the questions and the objective are somewhat different, although not completely different, it's somewhat different for all those aspects, Tom, you put there. So that's why the methodology could be slightly different, but they are all helpful. Then in terms of, the safety signal, Frank highlighted very well. Absolutely, it's in a ways we do need looking into the benefit risk there. It's also, based on the objective, right? And when we develop a drug, we look at, phase 1, phase 2 versus those pivotal, the most of the time it's a phase 3 studies there. The goal is different. In the phase 3 pivotal study for regulatory submission, we certainly very cautious about the false positive. If a drug is not working in certain area, we hope we will not say actually it's working, right? Because it's a false positive. But within the company, we also do not want to kill the drug prematurely. It's a false negative ones is over there. So I do think all those methodology are very helpful. That depends on the stage of the drug development and the stakeholder we work with.
Thank you, Chi. May we have the next slide, please? So there are several audiences interested in subgroup results. For example, prescribing physicians, patients, and drug developers, and of course, other researchers as well. Each group has specific interests and assumed levels of quantitative fluency. How should statisticians address the needs of various groups? So I'm really interested in this sort of question, and I have some opinions of my own I'd like to share on this. And some of this lines up pretty well with what Loka Chakravarti said in her opening remarks, and was sort of specifically around the patient snapshots program. And those the data should be available, accessible, and transparent. Data and more than the data, the information that's coming from the trials associated with the studies, the drug studies. So with that in mind, I'm also thinking about something that Dr. Ucker said that really stuck with me, and that was this infatuation with point estimates. So in thinking about these and how we as statisticians need to address different audiences, so drug developers, for example, interpreting information that's presented will have a higher degree of fluency and be able to think about the assumptions that are implicit in the models, whether they're Bayesian models or frequentist models, but there's always assumptions that go into the models. And we had some discussion about exchangeability, and whether it's exchangeability or other assumptions, the point estimate is presented to the end consumer. And if this is a sort of an unsophisticated person saying, well, that's the point estimate, now that's close to me and I'll believe it. And sometimes they may be presented with say the forest plot, and they can take the variance into account a little bit, but really focusing on the point estimate. Now when you put shrinkage into it, you see, well, there's several different ways to think about shrinkage. How is the end consumer going to recognize this, and given these different levels of fluency? So I think that presents a challenge for how do we to all these different audiences explain the fact that there is some amount of assumption and consideration going into this. And one of our speakers mentioned that, well, there's a human intervention in choosing assumptions and models to use. And does the end user have that and the impact it can have on the analysis? So I see these as challenges for us in how to present the information that we get. I think Dr. Griswold's graphs and his presentation was excellent. The scrolling storyteller, that concept is great. Implementing it, of course, will be resource intensive, I think, because a presentation for every product to go through that level of development would be a task. So along with the interpretation of plots and going back to this idea of understanding our standard forest plots are not always interpreted appropriately. Often people will look at the confidence intervals and do their own tests without considering multiplicity. It may or may not be appropriate. Are people comparing the confidence intervals to some benchmark? Are they looking at overlapping confidence intervals? Is this information clearly coming out? And they're usually presented without any explanation and thought to how they're going to be interpreted. There's an assumption that people interpreting them will have an understanding. So just to sort of round it out, that is you know, a focus that I would like to see addressed is how do we take what we estimate with our you know, variance estimators and then present it to people so they understand exactly what's gone into this. And you know, there could perhaps be only a few people in a subgroup, and there's the point estimate, and what does it mean? So to clearly have that concise point brought home with all the technicalities in it, that's a challenge. So I'll pass on the question to Frank and see if he's got something to add.
All great points, Tom. Not a lot to add. I think the discussions we had yesterday, I think Scott and Stuart and Steve Ruberg, and then followed on today by Michaels, are the patient is interested in, you know, what happens with patients like me, and largely the healthcare practitioner is going to focus on the same issue. What are the clusters of patients that look like me, and what is the benefit to risk of this treatment in that cluster? The developer is usually looking for as broad a population as possible, so they have a slightly different motivation, but they are, in all cases, want the drug to have a positive benefit to risk. I guess one of the questions I have is, are there areas, and I think that Don's example was good in this regard with his example of Brazil. Are there examples, if we have an overall effect, is heterogeneity of treatment effect is fine. I think as Stuart pointed out, as long as it all goes in the same direction. But how do we determine whether or not there literally are subgroups of patients where the treatment does not work or has a negative benefit to risk? And I think that the borrowing and shrinkage estimators today were extremely helpful. I think the example that was worked through on the life study in African Americans is another good example. And then that can determine from a regulatory standpoint how that reflected in the label. I think the graphical, I agree with Tom, the graphical process that Michael went through is excellent. And one of the things I like about it is it's transparent. So you can show the patient what happens to a patient like them, but you can show also what happens to a spectrum of patients around them, as opposed to, I agree, it's just a point estimate. So I think that the graphics are great as long as they can have integrity and transparency, because graphics can be abused just like a point estimate. But I think that the idea of looking at benefit to risk and making sure that you have this stems from the regional difference project that MRCT had that we talked about yesterday. Are there countries, regions, or subgroups where the treatment does not work and does not have the overall effect? And as I say, I think Don and Tom's presentation helped a lot in that regard today.
Would you like to add something?
Is that me or John? Which one go first?
Chi, go ahead.
Okay, thank you. So I'm not repeating anything Tom, you and Frank already said, all the great points there, along with what has been shared by two of you. Just a quick comment here to share is, as we all know, we develop a drug for our patients, then for physicians to give the drug to patients. So that means, you know, how to explain those in a simple way to physician, to patients become very, very important. So I think it's, you know, and we absolutely still need to continue use those simple methodology, because the community is so used to see those methodology, it's easier for them to understand, like a point estimate, et cetera. But nevertheless, there's a tons of more advanced, innovative methodology can really help us just to make sure the message we convey is solid. So in other words, regardless those point estimate or forest plot, et cetera, when we present those, also do more sophisticated methodology, including the Bayesian shrinkage estimate and including all those sensitivity analyses, and also we call it trigger point analysis, just to make sure all the methodology is very solid there. I noticed there's quite a few the chat change talking about, interactive graphic presented by Michael. Again, that was wonderful presentations over there. And yes, talking about, when non-statistician looking at those, they might come up, the message could be somewhat misleading. Most of the methodology all may have this issue because they would need people have a solid statistical thinking to understand. So that's why the guidance along the way will be very important over there as well.
Thank you, Chi. John, would you like to add?
Sure. I'm not sure how much of it will be adding versus echoing. Tom, I liked your framing of this question and your discussion about it a lot. I agree that these are really important, essential challenges, maybe some of the most important topics of the workshop. The idea of balancing transparency and the potential provision of misleading information is really hard. It is, I think, making information accessible and transparent is a truly noble, good goal in itself. We need to provide information that supports people making decisions based on their own values and priorities. But also, on the other hand, going back to sort of real basics, something that's discussed often on the first day of Stat 101 courses, is that data and information are not the same thing. And I personally, this is maybe a philosophical issue, but I don't think there is such a thing as value-free or neutral display of data. If you show someone raw subgroup data, there is an implicit rhetorical effect of saying this is important. And if there were only 3 people in the subgroup, it's not important. It really isn't. It's not interpretable in any way. So it is all of this is just to echo that it's difficult. I don't think there's a one-size-fits-all answer to these questions. I also wonder if the difference in quantitative fluency between potential audiences is as great as the question on the slide implies, or at least if it has the same effect. I would say if a patient looks at a forest plot that has 20 unadjusted confidence intervals and they say, I don't know what this means, and you compare that to a clinician who looks at the same 20 unadjusted confidence intervals and says, oh, this one that doesn't overlap 0, that's the one I'm going to pay attention to. I'm not sure the clinician got it more right than the patient did in that situation. Again, not to sound like a broken record for my answers to other questions, but often the most important thing is really giving an accurate impression of the uncertainty, because I think people's cognitive biases tend toward overbelief and overconfidence when they're presented with specific numbers. So whenever possible, I think we should be sharing our uncertainty as regulators or as medical practitioners with patients. Thanks.
Tom,
thank you, John. Thank you. Yes, please, Chi.
Yeah, because thank you, Tom. What John said reminds me something here. So I think another point to add along the community and the conversation here is the education and the communication. I remember both Aroga and Lisa and others mentioned, FDA site, right? The impact story. It's a very, very helpful. I look at the site just to pretend I did not know anything in the statistics. I absolutely fascinated about what's there. Talk about the methodology, the communication, all kinds of things. So it's an education journey, and the more we do something like that, and the more the patients and the physicians, they will understand more. Tom, we are not able. Okay, now we can hear.
Yes, I was. I have a problem muting myself or unmuting myself. Thank you for wrapping that up, and we'll move to the next question unless there's any more comments. So, does your organization use a standardized approach to HTE? Do you see any prospect for improving standardization? And I'd like to ask John to start off with this question.
Sure, I'll be happy to. Standardization may be an exaggeration, but we do have certain routine practices in statistical review at CBER, at least. We always look at the primary efficacy results by age, sex, and race, ethnicity subgroups. We don't necessarily do that with any great expectation of interpretable results or differences. We do it more as a matter of habit and to make sure we're not missing something really meaningful and important. We also very often will look at regional differences and at baseline clinical characteristics whenever they're scientifically relevant. And again, with regional differences, we might have a somewhat more, we might expect to see meaningful differences a little more. Beyond that, the HTE assessments we do tend to be more ad hoc and situation specific. Thinking about suggestions for improving standardization, the one thing that comes to mind, and it's probably on those checklists, there's been a lot of discussion in the past couple of days about pre-specification and what that means and how much of it there should be. I think a useful exercise at the design stage is for investigators to think about the subgroup analyses they're going to do, and before they collect the data, marshal the evidence for believing that that subgroup effect may or may not exist, including mechanism of action evidence, prior empirical clinical evidence, underlying disease process differences according to subgroups, and to use that to help interpret potential observed effects later. Because I think every statistician has had the experience of awe at the ability of scientists to tell a story that explains or excuses any observed effect once it's been observed, and to sort of try to be a little more dispassionate before the incentives kick in once the data has been observed could be quite useful to guide thinking.
Thank you, John. Frank, would you like to go next?
Yes, I would say my 7 years in academia told me very little of anything is standardized. So I don't, but I would say there are some common practices that I observe, part of the culture, I suppose. And I would say it falls under the heading of pre-specification, but not pre-specification in the sense of hypothesis testing. It's trying to, and this follows on directly from what was just said, to get the clinicians to think ahead of time about, well, if I were to think about a model trying to understand how this treatment effect is going to vary across a response surface, let's say, what factors are important clinically, and how am I going to set that ahead of time? So it's based on a lot of things that were just mentioned. So try to get people to focus on, well, these 10 things are really important as a clinician, and I take them into account when I prescribe medicine. Whether they turn out to actually be significant or related to the outcome or not may be irrelevant in the moment. And to try and avoid the situation where after they get the data, they look at something in table 1 and they go, oh, well, there's 10% here and 7% there. I need to adjust for that, or I need to look at that subgroup. So I think trying to get I'll spend a lot of time, it can sometimes take a couple of months to get people to focus on, and I would tend to view it as a model as opposed to per se subgroups, and try to get people to look at it in the broadest possible terms. So that would be and I'd say that's a pretty consistent culture in looking for heterogeneity of treatment effect in most of the trials that I work on.
Thanks, Tom. All the great points raised by all of you. So maybe just to add very briefly here is, Tom, you used the standardized approach, which is here in the company, we put together so-called best practice, all standardized, so moving forward. So in a ways, we put together best practice document, which covers design analysis and reporting aspects. subgroup analysis, including HDE, is a part of the documentation best practice there as well. So of course, after this wonderful conference, we will try to strengthen the best practice efforts over there. So we started from design stage. I think John mentioned quite a few potential subgroups analysis there at the design stage. In the company, in industry, not every study teams know which subgroup to look at. So very often you see cross industry and cross product teams, the number of subgroup and the content of the subgroup could be very different over there. So that's why the guidance over there is very helpful during the design stage. Then of course, the afterwards is the analysis and the reporting and how to interpret those. So all those, basic methodology plus the modelings we all put there is absolutely looking into all kinds of different methodology, particularly watch out the assumptions there and look at the sensitivity analysis to see how robust the results will be. So all those things are very important moving forward and continue to be the case.
Thank you, Chi. May we have the next slide, please? What statistical methods or combination of methods balance providing information to stakeholders with the goal of not overinterpreting or misinterpreting available sources? So, Chi, would you like to start off this question?
Sure, Tom. Maybe just add one comment here first. Is can chime in a bit more afterwards if needed. So I do think a Bayesian shrinkage estimate, or you call shrinkage estimate, it's one of the methodology has been used now and need to and can be utilized much more widely over there. And yes, there's a tons of methodology in frequency states there as well. So, because the end of the day, as we know, right? I mean, the presenter give very nice presentations to patients. They often ask is what is the benefit or risk to me or to someone similar to me? So in a way is how to borrow and where to borrow and how much to borrow in the Bayesian setting is very important. So all those and the thinking is critical over there. I do think that Bayesian methodology tells you the probability the drug works, and it's very informative on top of those more the basic summary statistics over there.
Frank, would you like to go next?
Yeah, so I'd say if you'd asked me this question 3 days ago, my answer would have been completely different than my posterior answer after sitting through. I've learned, been exposed to so much in the last couple of days, and not to repeat some of the things I've said or what Chi said. I was particularly taken by the presentation from Don and Michael about this idea of I refer to it as a slider, I guess, to oversimplify. But the idea of being able to present the data and give you both analyze and present the data in a consistent manner that gives you the full range of information. And I think I was also appreciated Tom's comments on the exchangeability assumption, which I must admit I was probably had more a priori concern about them, but I think his explanation and description of that in his work actually was helpful. Not that I agree with you, not that you would ignore it, but I think it's no different than make other assumptions we make in doing other analyses, and I thought that was actually quite constructive. So I think the overall the combination of the graphical methodologies and the Bayesian shrinkage estimator, if properly applied, and I think both Tom and Don gave appropriate cautions around that. And the other piece that I thought was the methodologies that were presented by Scott and Stewart looking at the benefit to risk approach, which were then highlighted in some of Michael's presentation, were things I certainly thought about as potential use, but they gave me a lot more food for thought. So I think those are particularly important and helpful in that if you're trying to think about precision medicine in any sense, that has to include the benefits and the risks. And I think those methodologies were extremely helpful in trying to display that.
John, would you like to share some thoughts?
Sure, relatively little to add on this one. I agree with what Frank said. I've been impressed by all the talks, but I think in particular some of the displays in Michael and Dawn's talks provide a really good template for how to approach presentation and communication of these kind of data. I did want to say one thing about forest plots, because we've seen a lot of them in the past couple days. And as a sort of curative for just raw subgroup specific estimates, I think they're good. But I do think depending on the kind of decision that might be made based on the observed subgroup effects, people should consider whether the confidence intervals they're displaying in the forest plots, in some cases maybe should be more like simultaneous confidence intervals, sort of the equivalent of a multiplicity adjustment, which would widen them a lot, and in many cases, I think appropriately.
Thank you, John. That's a great point. You know, I'll just add a little bit onto this. I thought, you know, I expressed earlier that I thought all the presentations were really interesting and informative, and I enjoyed very much the presentations. I think in as far as methods, I found the door method and the risk modeling, I think, are methods that sort of incorporate benefit and risk simultaneously and present that are very interesting methods. And I think, you know, if they can be presented to the, well, I'm thinking specifically of a patient's perspective, how they're presented to a patient to, you know, sort of look at their benefits and risks and help them with their personal decisions. You know, what is this drug going to do for a patient like me? And then, you know, incorporating this idea of how do you look at the trade-off? And I think it was put in the terms of partial credit, because everybody will have sort of a different perspective on importance of risk. But I thought those that sort of incorporate both the safety and you know, the benefit were valuable, and should definitely, those types of interpretations take more prominence than they currently do. Where currently we look at everything, you know, split out one-dimensionally. So we are just about at our allotted time, and with that, I would ask if any of the panelists would like to make some more comments. Yes, please.
Tom, if you like, we can spend another 15 minutes or so, because we'll have a rapid close after this discussion.
Okay, well, we have a few more questions I can offer to the panelists, and so let's see. Can we go to the next slide, please? So, without hypothesis testing, are there any differences between efficacy analyses pre-specified versus efficacy analyses not pre-specified? And should we perform corresponding safety analysis in those pre-specified subgroups on efficacy? For safety subgroups, should we look into different metrics, for example, relative risk, absolute risk differences? And there are quite a few points to unpack here. And I'd like to ask John to go first.
Okay. Sure. So in terms of the pre-specified versus not pre-specified efficacy analyses, again, outside of the area where it's, you know, like there's pre-specified hypothesis testing and alpha has been carved out or whatever. Ellis mentioned yesterday that he felt that there was no difference between these and that they were both exploratory. I half agree with him. I think they certainly are both exploratory. These kinds of analyses should generally be used to support further investigation rather than more practical decision making. But I do think that there is a difference between pre-specified and not pre-specified, in that if something has been pre-specified, there's a much higher presumption of mechanistic plausibility and or of importance of the subgroup difference. Generally speaking, anything that comes in that's a total surprise or was not discussed at all in the protocol, I would say mentally I downgrade that quite a bit in terms of how much weight I would put on it versus something that people were curious about, meant to look at, but perhaps didn't intend to confirm in a trial. So I would think it is important to retain that distinction. In terms of safety, safety is tough. It depends on the specific situation and the specific safety outcome you're looking at. If it's an adverse event of specific interest and it occurs at a high enough rate to make this kind of analysis meaningful, sure, it could be helpful to look at subgroup specific analyses. But many of the adverse events we're looking at either are unexpected or are not occurring at a high enough rate to believe any of the cell counts, much less differences in cell counts between groups. So usually no. Short answer, usually no.
Thank you, John. We've had a request from Ellis Unger to make a comment, and I think since we just brought up his name, I think that would be appropriate. So, Ellis, can you unmute yourself and comment?
Yeah. Thank you very much. So I will make this point that companies specify subgroup analyses maybe because they have a hypothesis, some mechanistic hypothesis, but they also specify subgroups because they have to. They may not think that there will be they may not predict a difference in response by age or sex or anything else, but they have to do those subgroup analyses. And so there's no hypothesis there. They may have hypotheses and add some subgroup analyses because of their hypotheses, but it's a mixture. So Tom, maybe you could respond to that.
So
I said Tom, I meant John, actually. Sorry.
Okay.
Oh, okay.
I'm sorry.
Yeah, no, Ellis, I completely agree. Yeah, it absolutely is a mixture. Going into most trials, we don't expect a sex difference. We certainly don't expect a race ethnicity difference in most trials. I was trying to hedge that a little by saying that the distinction would be either in plausibility or in importance, because if there was a true sex difference, presumably that would be very important to prescribers. But yeah, no, you're right. And if we can distinguish between plausible hypothesis-based, evidence-based subgroup analyses and those that aren't, that's really the truer distinction. I guess what I'm saying is the ones that aren't pre-specified are almost never the ones that seem most plausible. But completely agree.
Thank you, John. Thank you, Ellis. So I'd like to ask Chi if she'd like to contribute to this question.
Sure, absolutely. Thanks, Tom. So yeah, the points raised by John and Alice, I certainly agree. When I look at the one hand from registration point of view, from labeling point of view, regardless pre-specified or not pre-specified without the hypothesis testing, there's no multiplicity control adjustment over there, right? So they are very similar in many ways. On the other hand, is sometimes in the drug development journey, we may not be very clear. It's in a ways, some variables, parameters are prognostic factor. sometimes we just are not very clear if they are or they are not. Then when you talk about the ad hoc, post hoc analysis, could have tons of them for many endpoints. So when you do that, then you may see a lot of subgroup analysis, and those so-called order results by chance could occur. So I think best practice is, again, to go back to design stage, think about what kind of a subgroup should be there, then interpret it afterwards with caution. And in terms of your second bullet, it's always fascinating. I always felt very fascinated about this is, in a ways, as we know, not to talk about the subgroup analysis. John and Alice were referring to the age and the gender. Those are pretty, the clear, it's important over there. What I observed all those years is when we talk about the efficacy analysis, often there's many subgroups there, sometimes, for example, including ECOG, et cetera. And then we look into all those subgroup analysis, but I don't think people often look into corresponding safety subgroups over there. So come back to Frank mentioned many times, the Stu, Scott, and Michael, and Don, all the others talking about the benefit risk, right? So when we look at the subgroup, the benefits there, but we do not necessarily looking into the safety in the corresponding subgroup there for many reasons, as we know. And so I do think sometimes maybe it's helpful and look at both benefits and the safety, not as a default, it's just for certain subgroups which are relevant and looking into the both aspects. And the last point about the matrix and metrics is, of course, for the drug submission, we always, as we all know, we need to pre-specify, right, which one is a primary approach. So every, regardless relative risk or absolute risk or odds ratio or hazard ratio, whichever you talk about has its own pro and cons. But from clinical meaningfulness point of view, actually, I should say it's easier for people out there to understand that maybe absolute risk, it's the one, and it's easier for patients, for clinicians out there to understand. But nevertheless, I always think it's very helpful also looking into other aspects based on the relative risk, based on the odds ratio, just to make sure the signal you detected, identified, is comprehensive enough.
Thank you, Chi. Frank, would you like to add?
Just to add a couple of things. I'm just slightly different view than John, I guess. After my experience in safety, I would say actually many adverse events, adverse drug reactions actually are not unexpected. If you look at the biology and comorbidities and animal data, even though you didn't expect, maybe you were hoping they didn't happen, but they're not always that unexpected. So how you relate that to subgroup, I mean, I would say, if look at the first question, I would never look at efficacy without looking at safety, like turn that around, and assuming I don't have a numbers problem. If I have a potential for a safety signal, I would never interpret that without understanding the benefit. I mean, that's what you would do if you're on a data monitoring committee. And I think so I think those are, to me, they go hand in hand, and was some of the things I think Scott was trying to raise. But I do want to raise some of the excellent points made by Michael and Frank Harrell, Michael Griswold and Frank Harrell. I mean, I tend to like to look at the absolute risk, but their demonstration of that the fact the odds ratio may be actually more stable, I think, was a very useful exercise, and it's a good teaching moment. But one of the things that I have observed in people trying to look at whether it's a subgroup or overall is whatever measure you like, use the same measure for safety and efficacy. I think people, sometimes I see people use a hazard ratio for efficacy and then they do absolute risk for safety, and are very difficult to understand benefit to risk. And also use the same population. We didn't talk about that either, that if you're good, I think that in Stewart and Scott Evans' examples, I think they were very careful to make sure they're using the same population for both. I think that's something that's it's sometimes a weakness in the way people look at benefit to risk. But I think my main message is you should never look at efficacy without looking at safety and vice versa. I mean, there are rare exceptions. There are some adverse events that are so obvious, like Hy's Law or Stevens-Johnson syndrome or something. But in general, for most adverse events, you should also look at benefit at the same time.
Agree. I agree with those sentiments. May we have the next slide, please? So this is very specific to Bayesian shrinkage. A few mentioned Bayesian shrinkage estimators. Can this be used not only for safety subgroups? And maybe I'll ask Frank to start on this.
Well, a great deal of my education on shrinkage estimators has happened in the last 48 hours. So I don't claim to be an expert on that, but the more I thought about it, Steve Snapper and I had a number of discussions leading up to the meeting about this. I think it's fine. I alluded to this before. I like the concept of trying to rule out spurious outcomes, maybe false positive outcomes, and particularly because it's very hard to, even though adverse events maybe not be pre-specified, there's a lot more outcomes. So I think that's very useful. My only caution there is that often safety issues are not related to a single adverse event. They're related to a spectrum or even a syndrome or a combination of events, and looking across a system organ class or some other relation may be what's important. And so if you're constructing those shrinkage estimators, understand the actual outcome you're looking for. And if I wouldn't necessarily apply it to an individual adverse event, I'd want to understand what the actual safety signal is. And certainly if you're trying to do it as a screening tool, I think it has great utility. If you're trying to understand, if I'm looking at literally 100 adverse events, and only one of them is sending a signal, then the shrinkage estimator may help me. But if I have a small signal in 6 different adverse events in the same system organ class, I don't necessarily want to shrink that signal. So I think a sensible approach to it would be quite useful.
John, would you like to add?
I don't have much to add except to say that, again, and maybe I was actually trying to type a clarification, just to clarify what I was saying about the previous safety question as well. I certainly don't want to have been heard saying that safety analyses aren't important, more that it's not often realistic to ascertain heterogeneity in safety outcomes outside of common events or very large trials. And that somewhat goes over to this question as well. Yes, you can use these methods for safety subgroups by all means, but if many cells are empty, then the priors are doing a lot of work. And if your priors were set up right, what you're going to likely find is that there's not much heterogeneity because it would take, again, very common events to overrule that.
Thank you, John. G, would you like to add something to this?
Sure, maybe just to add 1 to 2 comments here. John, you just mentioned go back to question 6. I go back to question 6 as well, because 6 and 7 are linked. So I certainly agree with you, Frank. You mentioned that during the DMC, right, the data monitoring and looking at the efficacy is very important along with the safety. On top of that, though, is I think when you look at, for example, the label or the publications, etc., people often present a subgroup analysis in efficacy, but often you do not see the corresponding safety analysis results in those subgroups there. So in a ways, it is the question for our field is whenever people present efficacy, should we see the safety, the results over there, because the end of the day, it's a benefit risk, right? That's what the patients really care about is how about, myself, him or hers, and what are the benefit risk for that specific subgroups there. So corresponding to this question 7, it's the same thing is Frank, you mentioned, particularly Frank, you mentioned is you did not use this methodology before the meeting, and we all learned much more through this fantastic conference here. So my own journey is only use the methodology for efficacy. It worked out. It was extremely helpful. I've been thinking about utilizing the methodology for safety. So if you ask me, I absolutely think the methodology can be applied to both efficacy and the safety, and especially for certain safety analysis, for example, AOIs, every company has different name, is a special interest of adverse events. And along with the methodology, just to look at, what kind of safety information you have there, and potentially reduce the noise there among the other methodology. And absolutely is when we utilize this methodology, because nobody really knows, exactly how much borrowing we should do. So doing those so-called sensitivity analyses and looking into all those potential, the primary assumption, the model assumptions there become very important. Thank you.
Thank you very much, Chi. And that is the last question we have on our list. So I think on the schedule is an open discussion.
Thank you. And thank you, Tom, and thank you, Frank, G, and John, for a most wonderful discussion. Really pulling together after yesterday and anticipating today some of the major questions that were raised by everyone, and addressing them in a most thoughtful and comprehensive way. We have been answering the questions that have come in. I do want to take a pause for a minute. Could I have the next slide? And say we're not going to have a formal open discussion beyond what we just had. Both Sharday and I would like to really thank the planning committee for all the enormous work that went into this discussion. And particularly, let me just say, Frank Rockhold and Steve Stappen were essential colleagues in developing both the idea for this conference as well as the sort of frame and ability to pull this together. And Sharday and Mark Rothman and many others at the FDA really worked hard week by week to pull the right individuals into this discussion so that we could really advance the thinking. The fact that the experts in the field themselves learned something over the last 2 days really is evidence to me that this was a success, that we have a lot of work to do, but some of the directions for what approaches we need to take and think about together, as well as what research and sort of thoughts need to be put into this, have been illuminated by the terrific discussions that we've had over the last couple of days. And I just want to thank everyone once again, all the speakers, the planning committee, and my colleagues at the MRCT Center, Hayat Ahmed, Sarah White, for having put this together with me before I hand it over to Sharday for closing comments. Sharday?
Thank you, Barbara. And I will just echo your thanks. This has been a very, very informative meeting, and I just want to thank everyone that was a part of the planning committee. Of course, all of my FDA colleagues. I truly appreciate the presentations and the information that they shared, and all of the work that they did to make this meeting a success. And also want to thank everyone that participated and attended this conference. We really appreciate your time. And just to kind of bring us back to where we started, coming into this meeting, we really were hoping to gain a common understanding of the desired goals of subgroup analyses, standards for assessing effects within subgroups. We talked about also defining differences in effect between subgroups, Bayesian approaches, advantages and limitations of traditional and innovative models of subgroup analyses. And I think that we have really had extremely informative and thought-provoking presentations and really robust discussions. So I just want to thank everyone for all of your time and the expertise and information that you shared over these past 2 days. And hope everyone has a great rest of the week and stay safe and well. So thank you.
Thank you.